Surabaya.
Sparkling Surabaya, a masterpiece dance rises from Surabaya heterogeneous society and grow the background of urban arts, make a unique style with modern performance. On the dance motif varies from East Java dances with traditional music. Ladies and gentlemen, this dance depicts the identity of the people of Surabaya, East Java province. The agility of this dance illustrates the spirit of Surabaya.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. We are delighted to welcome you to the International Conference of Pharmacy and Health Sciences 2020, the third joint conference between Universitas Airlangga and University Sains Malaysia. Hosted in the City of Heroes, Surabaya, the Faculty of Pharmacy Universitas Airlangga. It is being a great honor for me, Dini, and my partner, Viro, to introduce and welcome each and every one of you in this international conference. Your Excellency, Rector of Universitas Airlangga, Professor Dr. Muhammad Nasir, Vice Chancellor, University Sains Malaysia, represented by Deputy Vice Chancellor, Academic and International of University Sains Malaysia, Professor Dr. Ahmad Farhan Muhammad Sadullah, Dean of Faculty of Pharmacy, Universitas Airlangga, Professor Junaidi Kotib, PhD, Dean of School of Pharmaceutical Sciences, University Sains Malaysia, Professor Dr. Habibah A. Wahab.
And also our distinguished speakers for today, Dr. Anda Apoteker Raden Roro Maya Gustina Andarini, Master of Science, Permanent Inspector from the National Agency of Drug and Food Control of Indonesia. Professor Dr. Habibah E. Wahab, Fellow of the Royal Society of Chemistry from School of Pharmaceutical Sciences, Universitas Sains, Malaysia. Associate Professor Kornkanop Inkaninan from Faculty of Pharmaceutical Science, Naresuan University, Thailand. Yiwei Wang, PhD from Anzac Research Institute, Concord Hospital School of New South Wales, Australia, and also Dr. Fatimatu Zahra binti Abdul Aziz, PhD from School of Pharmaceutical Sciences, University Sains, Malaysia. And last but not least, Profesor Apoteker Bambang Prayogo Eko Wardoyo, Magister Sains from Faculty of Pharmacy, Universitas Airlangga, Indonesia. And also, we would like to welcome all of the presenter and participants of International Conference of Pharmacy and Health Sciences 2020.
The International Conference of Pharmacy and Health Sciences 2020 is the third joint conference between Universitas Airlangga and University Sains Malaysia and is organized by Faculty of Pharmacy Universitas Airlangga in collaboration with School of Pharmaceutical Sciences University Sains Malaysia. The theme of this year's conference is Research Acceleration in Pharmacy and Health Sciences through International Collaboration. The conference provides a forum for exchange of information on natural product and related topics, as well as aims to build and strengthen scientific cooperation amongst research institutions.
Excellencies, distinguished guests, ladies and gentlemen, on this special event, we have several agendas as follows. The first one, the opening ceremony, and then the second one, the first speaker session, and then followed by the poster presentation for the first session, and then we will have a lunch break, and then after that we would like to proceed to our second poster presentation, and then followed by the second speaker session, and last but not least, the closing ceremony. Now, for a brief moment, we would like to invite you to sing Indonesian national anthem, Indonesia Raya.
And next, we would like to invite you to sing to the national anthem of Malaysia, Negaraku.
Maju
Thank you. Ladies and gentlemen, now we would like to listen several speech. The first one coming from the Dean of Faculty of Pharmacy, Universitas Airlangga. Ladies and gentlemen, please welcome Profesor Apoteker Junaidi Khotib, Magister Kesehatan, PhD.
Assalamualaikum warahmatullahi wabarakatuh. Welcome to Surabaya. It is a great pleasure to welcome all distinguished speakers and all participants to Surabaya in the International Conference of Pharmacy and Health Sciences 2020. Third joint conference Universitas Airlangga with University Sains Malaysia on October 27 till 28, 2020. This joint conference has a big theme, research acceleration in pharmacy and health sciences through international collaboration. Due to the coronavirus as a global pandemic and your safety in our priority, the scientific conference will be held through video conference. This international conference is one way to maintain our academic reputation and also research reputation. As we know, by disseminating our research to other scientists and academic, and the university will be highly recognized and perhaps highly cited. This conference covers topics of interest include complementary and alternative medicine, pharmaceutical analysis, pharmaceutical toxicology, pharmacology and biomedical sciences, natural product, drug discovery, clinical community pharmacy, and other related topics. This is also as a forum to facilitate academics, researcher, industrial practitioner, and student engaged in the scientific event. We expected that we can expand our international research collaboration, especially in the building research capacity in terms of talent of researchers, the abundance of the resources of research facility, and manage of research. This is the best way to work hand in hand to solve some global health issues, especially in pharmaceutical and healthcare field, and to achieve healthy and better quality of life. We would like to express our appreciation to organizing committee for the good team and their effort to prepare this successfully conference. Finally, we wish all participants have an enjoyable virtual meeting in Surabaya and can take all of part of this webinar well, and the sharing of experiences by experts and each other for result in a big access to provide best way for pharmaceutical research during pandemic. I hope all participants in Indonesia, Malaysia, and other parts of the world always maintain their health and stay healthy. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you to Professor Junaidi Khotib. And the next is the welcome speech from the Dean of School of Pharmaceutical Sciences, University Sains, Malaysia. Ladies and gentlemen, please welcome Professor Dr. Habibah A. Wahab, Fellow of the Royal Society of Chemistry.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. First of all, my praise to the Almighty that with His blessing, we are able to gather today in International Conference of Pharmacy and Health Sciences 2020 and Third Joint Conference, UNIA-USM. On behalf of the organizing committee, I would like to welcome all distinguished speakers, guests, and participants to this conference. Ladies and gentlemen, I would like to take this opportunity to express my gratitude to Faculty of Pharmacy, Universitas Airlangga, who opens up an opportunity for us to co-host this auspicious event. UNE-USM relationship in pharmaceutical sciences goes back many years, but it is not until 2007 we decided to take the relationship to the next level by organizing the first USM-UNE joint conference. There have been many exchanges and successes achieved with this collaboration, where we have successful research visit, joint supervision, as well as joint publication. In 2008, we also embarked on unprecedented joint socio-educational community outreach program with UNE for our undergraduate students. The program, Praktisiswa Mahasiswa USM UNE 2008, were organized by the two universities and attended by about 40 students from USM and about the same number from UNE. It was highly successful and brought great impact for the community as well as the students involved. The year 2020 has brought us again together. This is an interesting year indeed. The United Nations has declared 2020 as the International Year of Plant Health, and the whole World Health Organization designates it as the year of the nurse and midwife. However, for most of us, 2020 will be remembered for COVID-19 pandemic. The pandemic has created an unusual situation for all of us, and which will become a new normal for the world. Social distancing has become a new buzzword, and working from home has become a new trend in our academic culture. But as an academic, I feel we are now even closer, especially with the technology that is able to connect us to online learning and online meeting. And today, we are all connected through online conference where we will be exchanging the progress of our work, discussing about the progress of knowledge, research, and advances in the field of pharmaceutical and health science. For me, this is a new achievement for both universities, and hopefully we will overcome the physical social distancing with new normal online social media communication. Ladies and gentlemen, I would like to once again record my gratitude and appreciations to the Rector of UNE, Deputy Vice-Chancellor of USM, Dean and faculty members of UNER Pharmacy, faculty members of USM Pharmacy, and especially to all community members, speakers, and participants, and not forgetting to all who have made this event possible and successful. I wish everyone here a productive and enjoyable meeting in this online conference. Thank you very much.
Thank you so much for the warm remarks, Professor Dr. Habibah. For the next speech will be addressed by the Deputy Vice-Chancellor for Academic and International of University Sains, Malaysia. Ladies and gentlemen, please welcome Professor Dato Dr. Ahmad Farhan Muhammad Sadullah.
Assalamualaikum warahmatullahi and greetings from University Sains Malaysia. Professor Muhammad Nasir, Rector Universitas Airlangga Indonesia. Professor Junaidi Khatib, Dean School of Pharmacy, Universiti Airlangga Indonesia. Professor Dr. Habibah Abu Wahab, Dean School of Pharmaceutical Sciences, University Sains Malaysia. Distinguished speakers and guests, ladies and gentlemen. Assalamualaikum warahmatullahi and a very good morning to all of you. On behalf of USM, I'm very pleased and grateful that once again we have been given the opportunity to organize the International Conference of Pharmacy and Health Sciences for the year 2020, the third joint conference of Universitas Airlangga and USM. The theme of this event is the research acceleration in pharmacy and health sciences through international collaboration. As the theme suggests, this conference was held with the objective to accelerate research in the pharmacy and health sciences through the collaboration of both Universitas Airlangga Indonesia and University Sains Malaysia through the organization of this conference. In this conference, we will indulge with a number of valuable findings from various research topics, namely complementary and alternative medicine, pharmaceutical analysis, pharmaceutical technology, pharmacology, and biomedical sciences, natural product, drug discovery, clinical and community pharmacy, and others. It has been well known that research acceleration can be achieved through collaborations and sufficient communication on the results and discoveries. Thus, I hope that this event will be an excellent platform for all participants to engage with the works from all speakers and presenters, as well as to get to know each other, foster existing and future collaboration. And for this, I would like to put this proverb: if you want to go fast, you go alone. But if you want to go far, go together. I would also like to add, not only that we need to accelerate, to catch up on things, but under the current circumstances, I think all of us researchers, academics, we must also be adaptive so that we can create more opportunities from the challenges that we are facing. Ladies and gentlemen, I was told that the history of this joint conference had begun in 2007, when the first conference was hosted by the School of Pharmaceutical Sciences of University Sains, Malaysia. It was then followed by the second collaboration in 2009, hosted by Universitas Airlangga. Even though the last conference was more than 10 years ago, I'm very delighted that we could rekindle this effort, and this joint conference has once again been reinforced. And I do hope that this will be the sign that we will re-establish and reconnect, and hopefully the strong bond between Universitas Airlangga and USM will continue to be stronger and be sustainable. It has indeed been a challenge in the beginning, where we were faced with the COVID-19 pandemic situation, that we are still facing the situation today and making the physical conference impossible. However, together we will overcome the challenges, and inshallah through this online conference we should successfully achieve all the intended outcome from the knowledge and also from the networking that we will establish. And with that, I would also like to take this opportunity to express my appreciation to the Director of Universitas Airlangga, respected Professor Muhammad Nasir, who I'm sure recognizes this strong relationship and supports this momentous event. I would also like to thank and congratulate all the committee members from both universities for the hard work, dedication, and I'm sure a lot of creativity and new ways in actually organizing such conferences. With all the challenges, I'm sure the feeling of satisfaction will be there, and I'm sure we will be very, very enriched by the conference. We hope that with this joint conference, our relationship between the two universities will be more dynamic and stronger in the future, and we can also make new friends and new collaboration with our other colleagues. Finally, to all participants again, enjoy this new experience. I would like to express my warm welcome to all of you to this conference, and hopefully you will have an enjoyable and unforgettable virtual conference with new insights, ideas, and partnership. May it also bring stronger ties and also elevate our prominence in terms of the knowledge, also the research work that we will do from now on. With that, I wish to thank all of you again. Wabillahi taufiq wa hidayah. Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you to Professor Dato Dr. Ahmad Farhan. And now we're going to listen to the welcome speech given by the Rector of Universitas Airlangga, Indonesia, and also officially opens this conference. Ladies and gentlemen, please welcome Professor Dr. Muhammad Nasih, Sarjana Ekonomi, Magister Teknik, Akuntan.
Honorable representative from School of Pharmaceutical Science, University Sains, Malaysia, honorable speakers from the National Agency of Drug and Food Control of Republic Indonesia, the School of Pharmaceutical Sciences USM Malaysia, the Faculty of Pharmaceutical Sciences Naresuan University Thailand, and ZAC Research Institute, Concord Clinical School, Sydney University, Australia, and also the Faculty of Pharmacy, Universitas Airlangga, Surabaya, Indonesia, honorable presenters, researchers, and students. Assalamualaikum warahmatullahi wabarakatuh. Good morning and a very warm welcome for all of you at the International Conference of Pharmacy and Health Sciences 2020. It is with great pleasure for Universitas Airlangga to be able to organize the third joint conference together with University Sains Malaysia. The conference provides a forum for the exchange of information on natural products and related topics, as well as aims to build and strengthen scientific cooperation amongst research institutions. University Sains Malaysia has been Universitas Airlangga's strong partner globally. Hence, we are very grateful to have this event together. Ladies and gentlemen, we welcome academics, researchers, industrial practitioners, and students to participate in the discussion on pharmaceutical sciences that range from wound healing and the use of medicinal plants, traditional and complementary medicines, to the growing use of herbal medicines. In conjunction with the conference, pre-conference workshop also available covering two topics, such as computer-assisted drug design, CEDD, and SPTLC, fingerprinting as a quality control of herbal product. The use of herbal medicinal products and supplements has increased tremendously over the past decades, with not less than 80% of people worldwide relying on them for some part of primary healthcare. It is important to put together the knowledge, skills, and practices based on the theories, beliefs, and experiences on complementary and alternative medicine. All this, whether explicable or not, shall be used in the maintenance of health. Not just that, it shall be used as well as in the prevention, diagnosis, improvement, or treatment of physical and mental illnesses. Pharmaceutical analysis and pharmaceutical technology need to be expanded to answer the challenge, because in the past, many rely exclusively on experience or observation handed down from generation to generation, verbally or in writing. The popularity and evidence of continued research use base clearly indicates that there are still lessons to be learned from traditional practices, especially regarding pharmacology and biomedical sciences. Although natural product drug discovery has shown promising potential in the practice of herbal products, many of them remain untested and their use is either poorly monitored or not even monitored at all. Therefore, the development of modern scientific medicine is still needed to be explored, which might include clinical and community pharmacy. Ladies and gentlemen, we do hope that this third joint conference with University Sains Malaysia can benefit society at large, especially in the study of pharmaceutical sciences in pursuing and explore combinations that could have a place in contemporary scientific medicine. The conference is also part of our program to increase the number of international research and publications, and therefore it's important to establish an international collaboration that leads to international publication. Let's not forget that we need to continue establishing the global academic community through the development of leading, innovative, and independent international activities like this conference. For University Science Malaysia, we do expect that this collaboration can be strengthened and even more be sustainable for years to come. Thank you very much for your kind attention, and I hope that you will have a fruitful discussion. Assalamualaikum warahmatullahi wabarakatuh.
Thank you so much, Professor Muhammad Nasi, for the opening remarks. Ladies and gentlemen, now we will hold a photo session via Zoom. We are going to have a photo session. The first session, we would like to ask all of participants in the Zoom meetings, includes all panelists and distinguished guests. Please kindly turn on your video and take a position in front of your Zoom screen. And the committee will take a picture. Please show us your best smile. Be ready, everyone. One, two, three, smile. Thank you. And for the next photo session, especially for the VIP member, please, I would like to invite Rector of Universitas Airlangga, Deputy Vice Chancellor of University Sains Malaysia, Dean of Faculty of Pharmacy Universitas Airlangga, and also Dean of School of Pharmaceutical Sciences University Sains Malaysia, all of the moderators and the speakers for the speaker station, the chairman and co-chairman of International Conference of Pharmacy and Health Sciences 2020. To other participants, please kindly turn off your camera as we would like to take a group photo of the VIP member. One, two, three. Give a smile, say cheese, or say something. Thank you. We have a great photo session.
Okay, distinguished ladies and gentlemen, now we have reached the end of our opening ceremony, and that was only the beginning. Before we start with the first speaker session, there are some announcements regarding this conference. The first, the abstract book can be downloaded from our website, and the link is already shared in the chat box by the committee. The participant who is entitled to an e-certificate should meet the requirements such as fill out the attendance list no later than 3 p.m. Western Indonesia time at the following link, as you can see on your screen and also in the chat box. And the second, participant should fill out the questionnaire form at the following link on your screen. And the participant have already submitted the certificate payment receipt. And for the participant, if you have any question to the speakers, please write in the Zoom Q&A box with format as follows: name _ ask underscore speaker's name underscore the question. For example, Neni Purwitasari underscore ask underscore Maya Gustina underscore what is the impact of the latest regulation regarding herbal medicine on herbal startup business?
You may find out all of the information with regard to the announcement in the chat box on the Zoom. Thank you, Ms. Dini, for the wonderful explanation. Excellencies, ladies and gentlemen, now we've come to our main agenda, the first speaker session, who will be led by our moderator, Professor Dr. Apotheker Mangestuti, Magister Sains. First of all, allow me to introduce our moderator. She is a professor in pharmaceutical botany and pharmacognosy at Faculty of Pharmacy, Universitas Airlangga. She has research interest area in alternative and complementary therapies for women's health, aromatherapy, Indonesian and other Asia's traditional medicine or ethnomedicine. And last but not least, he also has interest in medicinal plants with anti-osteoporosis and neuroprotection property. Ladies and gentlemen, without any further, I have the honor to hand over the session to Professor Dr. Mangestuti. The time is yours.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Good morning, Honorable Rectors, Vice Chancellors, Deans, Speakers, participants of the conference, distinguished guests, ladies and gentlemen. I would also like to welcome all audience to this international conference on pharmacy and health sciences 2020. It is my pleasure to be the moderator for the plenary sessions. And for this session, we will have 3 prominent speakers from Indonesia, Malaysia, and Thailand. Discussions, of course, will be held after or at the end of the session, except for the first speakers. So audience, please don't miss this opportunity. Write your questions using Q&A icon, and the format is name underscore ask, ASK underscore speaker's name. Let me start by sharing a short profile of each of the first speaker. The first speaker of this session is Ibu Dr. Randa Apotheker Raden Roro Maya Gustina Andarini, Master of Science. She is a pharmacist, pursued her postgraduate study on molecular toxicology at the Department of Pharmaceutical Sciences, Vrije Universiteit Amsterdam, the Netherlands. Afterward, she participated in various continuing education and technical training organized or in collaboration with Vrije Universiteit Amsterdam. She also participated in the training on safety assessment of cosmetic in the EU training course 2010, Vrije Universiteit Brussels, Belgium. Her present position is Deputy Chairperson of the Traditional Medicines, Health Supplement and Cosmetic Product Control, the National Agency for Drug and Food Control of the Republic of Indonesia. Her presentation is entitled Traditional Medicine Regulation, Strategic Role of Indonesia Food and Drug Administration in Supporting Traditional Medicine Development. Assalamualaikum. Good morning, Ibu Maya Gustina.
Waalaikumsalam warahmatullahi wabarakatuh, Professor Mangestuti. Good morning.
Good morning. Thank you for being with us today. We are all curious to know what is on the traditional medicine regulation of Indonesia. So you have 30 minutes for the presentation, followed by 10 minutes discussion. I will give you a reminder 5 minutes before the due time. So let us give a warm welcome to Ibu Maya Gustina, and time is yours.
Thank you, Prof. Mangestuti. Good morning, everybody. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to say thank you for the committee who inviting me as a speaker here. But maybe I would like to revise that now my position is inspector in Badan POM Indonesian FDA. Recently, my job is deputy chairperson of traditional medicine, hair supplement, and cosmetic product control. Just 3 weeks ago, I have been replaced in my new position, but as my experience during working in Indonesia FDA, I have been maybe 26 years. I work for traditional medicine, health supplement, and also cosmetics. So I hope my knowledge still can be shared for everybody and give overview about what the policy Indonesia FDA to handle the traditional medicine and to support herbal in Indonesia. And also you can see that we really encourage the small medium enterprises to do the business in herbal medicine. Let me to share my presentation here. Maybe can help to understand. Okay, what I would like to say. So my title is Traditional Medicine Regulation, and I would like to share what the strategy of the Indonesian FDA to support herbal medicine in Indonesia. So moreover, during the pandemic, you know that everybody have awareness that herbal medicine can be support of our immunity. Yeah? Besides, maybe we can use health supplement. But you know that health supplement, mostly the raw materials are imported from abroad. So if we want to do something ourselves, we can propose the herbal medicine as a health supplement, as immunity supporting during the pandemic. So this is my outline. Okay, and for introducing first that our president have vision and mission, and some of the mission of our president related to our responsibility in Indonesia FDA, such as the first, the increase of Indonesian human quality; second, competitive, productive, and independent economic structure; and the point number 7, protection for all nation and provide security for all citizens. And the next, the management of clean, effective, and trustworthy government. And the last, synergy with the local government in the frame of unitary states of Indonesia. This is the mission of President, and we implemented it in our policy in Indonesian FDA. So for the give overview that this is organizational structure of Indonesian FDA. Below chairperson, we have two administration section. The first, principal inspector. This is my job now, and then permanent secretary, and we have four deputy chairperson for the technical. The first is deputy for drug and narcotic, psychotropic, precursor, addictive substance control, and deputy for traditional medicine, health supplement, and cosmetic control. The third, deputy for processed food and product control, and the last, deputy of law enforcement. This is the new deputy in our new organizational about 3 years ago. Yeah, this is recently for the deputy of law enforcement, only for the echelon 2, yeah, echelon 2 in Badan POM, now become echelon 1. So from the presidential vision and mission, we have vision that the safe drug and food to enhancing public health and national competitiveness. So beside of our responsibility to protect the community from the drug and food which are substandard, we also encourage that the good better the good drug and food can be increase the national competitiveness. And our missions are to facilitate the development of drug and food industry, in particular micro, small, and medium enterprises, to strengthen drug and food control system to ensure its safety, quality, and efficacy, to increase partnership with other ministry, agencies, intergovernment, academician, societies, media, and citizen involvement in drug and food control, and to strengthen the drug and food control organization and human resources capacity. This is our mission, and this is
Hey, buka Fa.
you can see that Badan POM, we have full spectrum control, especially for traditional medicine. We do the certification of production facilities as a GMP certificate for traditional medicine, and the product before marketing, before distributing to Indonesian area, they have to get the registration number, and we will evaluate the safety and quality of the product first. And also the advertisement of the herbal medicine, we also do the evaluation before they launch the advertisement to the media. So we call it pre-market control. And then after the product go to the market and during distribution, we also do post-market control as a sampling and production testing, and also inspection to production facility and distribution. And we also watch the advertising and labels, and also we do the pharmacovigilance. So we assure that the consumer can get the proper drug and food in the market. So we control the safety, efficacy, and quality of the traditional medicine. Besides it, we do also communication with the consumer. Communication, give the information, and educated them how to protect themselves from the inappropriate food and drugs. So that's mean the community, our consumer can have awareness to choose which the product can be used, can be taken. So if something wrong during the post-market control, we do the administrative sanction, recommendation, also investigation, and go to law also if something too hard, yeah. And what the Indonesia traditional medicine potential. So you know that in the world we know about the herbal medicine in the worldwide, something like traditional Chinese medicine, Ayurvedic in India, also Kampo in Japan, and Indonesia we have Jamu. So Jamu is a language from Java, Jamu. So this is our icon that traditional medicine in Indonesia, we call it Jamu as empirical history. So it is also possible to get the herbal not only from empirical and also the new research. So they can do until clinical study if no empirical study before. So WHO estimate that the herbal use in the world reach 4 billion people, and now we can see that in the world they have encouraged to trend back to nature and increase the herbal use. So the commitment back to nature can increase the herbal use in the worldwide. So in Indonesia, we have so many herbal. I think the species in Indonesia more than in China. So now this is the good moment for researcher to do something to increase the herbal use in Indonesia. So we don't have to import the raw materials from abroad for the health supplement, for example. So herbal can be used for curative, rehabilitative, and promotive prevention. It depend on the evidence base. So it will be nice if natural medicine can be used in health services, and of course will be achieving faster health development. So we have overcoming multiple burdens in health development due to the emergence disease and non-communicable disease. Based on Indonesia Riskersdas primary health research in 2010, there are 59.12% of the people who were used herbal medicine, and 95.6% admit that herbal medicine is beneficial. So this is a good point for the using of herbal medicine. We can see that Indonesia is very rich with the biodiversities. Of course, is included sea biodiversities. We have so many herbal can be used for remedy, for medicinal, etc. Yeah? So depend on our research in herbal future. So this is sequences how herbal medicine can be used from natural until bioactive fractions. We have more technologies now and possibility to do research of the herbal medicine, and Badan POM Indonesia FDA always encourage the research, and also we will do side by side to do the research, and we will guide we will do the guidance of protocol of the clinical study. So this is the acceleration of natural drug research by Badan POM. So you see that from scientists go to in vitro and in vivo, and then the researcher will do the formulation, scaling up, and the researcher can be granting of the GMP certificate. And for the site of clinical trials protocols, these protocols should have acceptance from the Badan POM. So we will do together with the researcher, and we can prepare together, yeah, for the proper protocols which will be used for the clinical study. And then after approval from Badan POM and also ethic committee, the clinical trials can be done. So in phase 1, phase 2, phase 3, yeah, and afterward we will do the result of the clinical trials, and we evaluate it, and we have some of expert panel, and after we have we do approval with the product. Product can be produced with a commercial scale. So nowadays we know about the new term of the herbal medicine called OMAI, Modern Indonesian Herbal Medicine, or Obat Modern Asli Indonesia. So what the meaning of OMAI? OMAI we can equal it with the herbal medicine standardized of herbal medicine and also phytopharmaca. So what the opportunities of the OMI, the first substitute for lack of the conventional medicine, example, research on herbal medicine for COVID-19. So now we have about 14 clinical study under running in some of the hospitals in Jakarta, in Bandung, and some others, and they do the herbal clinical trials for COVID-19 patient. So the claim will be immunomodulator. And OMI can be used as a complementary of conventional medicine, for example, maintaining health for COVID-19 patient, and also as adjuvant on top conventional medicine for the people with comorbid of diabetes mellitus, for example, hypertension, and also, for this side, the patient should consult with the doctor for using this product, yeah. And challenge, there are a lot of researchers of traditional medicine, they have not been produced as commercial product, yeah. So we hope that the researcher can do the research which also can be implemented for the consumer or patient. So what the strategies for that? We do forming a task force for the acceleration of development and utilization of phytopharmaca as keputusan Menko PMK. So, and also encouraging the revision of regulation of Minister of Health number 55 of 2018 concerning the formulation application of the National Formulary atau Fornas and in the implementation of National Health Insurance Program, so that OMIs can be included in Fornas. Now, herbal medicine cannot be used for the BPJS, yeah, or insurance, national insurance, because not included in national formulary. So if include OMI into JKN, maybe the using of phytopharmaca can be broadened. So and then we can use our own medicine for the national insurance, and also encouraging the OMI in the national health system. This is maybe strategy of the OMI can be used for the formal health system. And the next slide, I would like to say about the importance of regulation. So, of course, regulation can be used for the imbalance information between the enterprise and manufacturer and the consumer. So we protect the misleading information, misleading of promotion. You see that the promotion of the herbal sometimes is very overclaimed. So this is not good for the consumer. It's not educated for consumer because of the misleading claim. So that's why we need the regulation to protect them. And of course, we protect the consumer, and consumers should have the good information and the balanced information about the product. So of course we protect of abusing dominant position, and also availability of public services, education, health, energy, etc. So this is why regulation is important in herbal medicine. And critical point of natural medicine quality assurance, because of collecting and post-harvesting process, it's not easy to do the continuously of the harvesting process, and also biological material variation, because you know that the same of the herbal if planted in Java maybe will be different if planted in Sumatra, for example, because of different of the environment. So the complexity of the composition and unknown bioactive compound extraction process also sometimes have problem, and chemical content variability, chemovariety, cultivars, and also analytical methods and standard are not available yet, maybe, and possible contamination. Moreover, in the small medium enterprises, so they have very simple technology, and sometimes they cannot make raw material properly, so they need our help, yeah, for government help to make small medium enterprises can produce the product, good product. And why the regulation is important? Because I said before that we protect the consumer and also to ensure fair trade. So this is why Badan POM or Indonesian FDA prepare some of regulation. So we have regulation about the industrial license, GMP, for registration, for post-market control, classification and labeling, clinical trial, toxicity test, implementing temporary protection, public service, and law enforcement. And all of our regulation can be downloaded in our system, our website called jdih.pom.go.id. Everything can be downloaded, all of our regulation, but still in Indonesia, so we don't have any translation in English. Maybe further we will do translation of our regulation. So now still in drafting of regulation for pharmacodynamic test, advertising control, and also side effect monitoring, and traditional medicine GMP, small and micro enterprises. So we do the slightly different GMP point for the small medium enterprises and the big industry. And this is some of the regulation of traditional medicine in Indonesia. I will not to say one by one, but you can see it in our website. Yeah, GMP, classification of traditional medicine, toxicity test, clinical trial, and market control. So some of the regulation already issued by Badan POM, and also the law enforcement if something happen, yeah, during marketing or concerning to the consumer protection. So in Indonesia, what the traditional medicine classification? As I said before, jamu is traditional medicine based on empirical history. And nowadays we have more than 11,000 products already give the marketing authority number and only supporting by empirical data. And the second class of the traditional medicine called standardized herbal medicine. And now we have 71 products. And what called standardized herbal medicine? This product already standardized raw material and products, and also already do the preclinical testing to animal. So after finishing those kind of the test and standard, so we can give the standardized herbal medicine, and in Indonesia we call obat herbal terstandar. And the third classification is phytopharmaca. Now we have 24 products. Why only 24 products nowadays? Because of phytopharmaca already done with the clinical trial. And we know that clinical trial is not easy, and so maybe quite expensive, yeah, for
Ibu
running
Maya, excuse
pharmaca.
me,
Yes.
you have
Oh.
5 more minutes.
Okay. Yeah, thank you. So we already have 24 product finished with the clinical trial. Now we have 14 product undergoing to clinical trial. Okay, and then this is marketing authorization, only the how to give the number for the product, yeah. And this is how to registration of traditional medicine. You can maybe read afterward. I will skip it. And so our regulation Indonesia in number 32 2019, we have a safety and quality requirement for traditional medicine. And the point is for raw material and also finished products. So you can see it afterward and in our regulation. Okay, Indonesia also include in ASEAN harmonization for the traditional medicine and health supplement. Yeah, this is the guideline. In ASEAN, we have already done with the discussion and preparation. So will be signed in 2021 and will be implemented in 2026 for the ASEAN harmonization. So this is my final remarks that Indonesia is the second biggest country in the world for its biodiversity and possessing high herbal natural resources. So many these herbal are used in medicinal treatment. And Indonesian FDA very encourage the development of the product, included the OMAI, and BPOM promotes the increase of OMAI by providing assistance starting from the preparation of test protocol up to the implementation of both clinical and clinical trials. And the existing regulations have been established to ensure the safety, efficacy, quality of traditional medicine, and some new regulations will be developed for facilitate the roadmap of traditional medicine and support OMAI to be accepted and into public health services. I think this is my presentation, Prof. And I'm so sorry if not complete, but everybody can see and can read my presentation afterward. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for a very interesting topic. Some highlights on Ibu Maya Gustina's presentation is Indonesian Food and Drug Authority has encouraged the development and use of OMAI, modern Indonesian herbal medicines, which is now categorized into standardized herbal medicines and phytopharmaca. Both have to follow government's regulation by implementing preclinical and clinical tests. Currently, various efforts are being made to encourage Obat Modern Asli Indonesia or OMAI to be included in the GKN, Jaminan Kesehatan Nasional or National Health Insurance Program. Therefore, scientific research is needed in order to develop safety, quality, and efficacy of herbal products. Nowadays, some new regulation will be developed to facilitate the application of roadmap of traditional medicine development and support OMAI to be accepted and used in public health services. Now it's time for a discussion. So we are waiting for questions from the audience. But I would like to start with by asking Ibu Maya Gustina. Ibu Maya, you mentioned earlier that communication with consumer is one of the important things that have to be done to protect consumers. So can you please elaborate more about how do you communicate with the consumers? This is very important.
Yes, thank you for the good question, Bu Mangestuti, because we realize that if we only do the control the product in the market, that's not enough, because consumer can they don't know about what the product, the product which already registered in Badan POM, for example. Moreover, now the marketing of the product through online, yeah. If they buy product by online, they will have difficulties to see the complete label, for example. And then we communicated with them. For example, we have something like kampanye cerdas menggunakan, or how to call in English, so campaign how to use the good product, yeah. Not only for herbal, also the others commodity which controlled by Badan POM. So we do something like give the clue. For example, we have click is kemasan label izin edar and kedaluarsa atau kemasan consumer should see the product. This is already broken or not, yeah. And then the label should be complete with the how to use it, direction, side effect, and then who is the the industry, who is the imported, and then also the number of the registration. They have to see it before buy. So
Okay.
how to buy from online? If difficult to see from the pictures in online, they can do chatting to the seller, asking to the seller, is it already registered to Badan POM or not? So we say that we always say to them that you spend the money, and then it will be so nice if you get the not registered product, for example. So from the simple things, I think consumer can be increase the awareness. So because they have to protect themselves from the bad products, not good product, not registered product, for example, like that. With many ways, we can go to them, to the school, to the campus, or we invite them to in our seminar, or we do webinar. So many things we can do.
Very interesting. There is a question from Fanya. Her question is there any regulation to promote or to encourage the use of OMAI to be prescribed by medical doctors? Dr. Maya, Ibu Maya.
Yeah. So nowadays, we don't have any policy to do that, but we communicate with some ministers, for example, Pak Erick Thohir, Minister of BUMN, and then also Minister of Health, and because this is not our responsibility to do that. This is under Ministry of Health.
Okay.
We only encourage them because phytopharmaca already finished with the clinical study, and then why not? It can be they use in formal health services. Why not? Because this of phytopharmaca already done with the clinical trial. This is the same with the conventional medicine. So it will be depend on the user. User is medical doctor, of course. And then we hope Ministry of Health have some program, some policy, some regulation will encourage this. Yeah, Badan POM only responsible for the safety, quality, and of the product. So we cannot encourage the end user or medical doctor to use it. This is a Ministry of Health.
Okay, thank you. Oh, so many questions. Next question is from Ermi. Her question is, do nutraceutical products or food supplements from herbal extracts produced from green extraction procedure using non-organic solvents still need complete standardization, such as OHT or phytopharmaceutical drugs? If it must be done, how the standardization procedure meet the BPOM regulations?
Okay, so basically that the product will be classified as a OHT, standardized herbal medicine or phytopharmaca. They have finished with some of the requirements. Yeah, for example, standardization of the raw materials and also they have done with the preclinical trials for OHT and clinical trials for phytopharmaca. If they want to do that, they can register the product as a jamu, as a herbal, as usual, not standardized herbal medicine and not phytopharmaca. We have three steps. Yeah, depend on the industry which one they want to choose. Yeah, maybe here Pak Tepi as a director of standardization, maybe you can give more information about that if the industry don't want to do something like a standardization and what happened for the registration. Pak Tepi is the director of standardization. Maybe Bu Mama Suti.
Yes.
Yeah.
Okay, thank you very much, Dr. Maya. Okay, I will explain a little bit about the standardization for herbal medicine. Yeah, because for past like the standardized herbal or also for the phytopharmaca, yeah, there is important thing for the standardization. And we have the Pharmacopoeia Herbal Indonesia, yeah, including so many medicinal plant, yeah, and the parameter, yeah, can do for the standardization for the herbal medicine. Yeah, I think that every country they have the pharmacopoeia. Yeah, so this is the handbook for standardization for herbal medicine. And if you have a new plan, yeah, you can prepare the document how about the standardization, and you can submit to the Baladan form. Yeah, and we can evaluate, and we maybe do something, yeah, testing in laboratory before we approve the standardization of your herbal medicine. Yeah, I think we have and every years we have developed this pharmacopoeia herbal for Indonesia herbal. Yeah, this is and every year we publish the supplement of this pharmacopoeia. Thank you very much.
Thank you very much, Ibu Maya Gustina. I'm afraid that we don't have much time. Yeah. So, but in conclusion, I might say that government from all Asian countries especially always gives strong support to the application of traditional medicines, also in Indonesia. So let us collaborate for the future of the benefits of in using it. Thank you very much again, Ibu Maya Gustina, and hope to meet you again sometime.
Thank you.
All right, distinguished guests, ladies and gentlemen, now is the second speaker of the session, Professor Habibah A. Wahab. Let me share with you the short profile of Professor Habibah. Professor Habibah obtained her PhD in pharmaceutical technology from the University of London in 1999. She joined USM as a lecturer at the School of Pharmaceutical Science, where she found a research group, this is important, Pharmaceutical Design and Simulated, which focuses on research on drug discovery, especially those utilizing computational chemistry and computer-aided drug design approaches. In 2012, Professor Habibah was appointed as Director General of Malaysian Institute of Pharmaceuticals and Nutraceuticals, Ministry of Science of Technology and Innovation, where she initiated My Nature 5000. What is that? A central natural product library modeled after USM Riken Joint Laboratory for Bioproduct Discovery, which she previously led. Since 2004, Professor Habibah has been an associate editor for Journal of Chemical Information and Modeling, that is an American Chemical Society publication. Presently, she is the Dean of School of Pharmaceutical Sciences, University Sains, Malaysia. Her presentation is entitled Natural Products for Drug Discovery in the 21st Century. Assalamualaikum, Prof. Habibah. Good morning.
Waalaikumsalam, Ibu.
We are very honored to have you in this session. We all know that at the present time, your topic of presentation is very important related to the drug discovery. So we know also that developing drug is a very complex work. So let us watch Professor Habibah's presentation to find a way out of it. Professor, you have 30 minutes and I will give you a reminder 5 minutes before the due time. Ladies and gentlemen, please welcome Professor Habibah.
Assalamualaikum warahmatullahi wabarakatuh. Thank you, Madam Moderator, Ibu Professor Magastuti, for the kind introduction. I think I am just as nervous as I am going to give a talk in front of a live audience. But anyway, let me begin my talk today by beginning. Good morning to everybody and thanking the organizer for the invitation to give a talk today. It has been more than 10 years since we last gathered in UNIA USM Joint Conference in Surabaya. I remember vividly on the opportunity speaking in the conference, and I remember much about UNIA hospitality and the privilege for me as a young lecturer to interact with the rector at the time, the dean, Professor Ahmad Sharani, and all UNIA faculty members. I remember that quite clearly. I would like to thank UNIA again for the great memories, and again today, I would like to, I feel a little bit nervous for some reason, I don't know why. It's virtual anyway, but anyway, although we could not be present physically to see our long-time friends, this online meeting is actually a great honor for us indeed. Ibu Dr. Maya just now explained to us about the regulatory aspects of herbal medicines in Indonesia. My talk today fits nicely on the development of herbal medicines or natural products. In my talk today, I would like to share some of our works on natural products and computer-aided drug discovery. I would like to apologize for the slight change of the title, but I hope we all get the message that this is also very important, right, to understand what we have been working on, right. I would like to share the audience this beautiful picture that I took from the internet by actually blend these two. So this is a picture of a slide of the picture of a royal balloon forest in Malaysia and Mount Bromo in Indonesia. I blended it with the conference logo. I thought this up to show how beautiful our two countries are. Not only that we are rich in our mega biodiversity, but we certainly rich in the knowledge of how we use our natural products. Right. Ladies and gentlemen, the contribution of natural products to drug development has been extensively documented. They still hold out the best options for finding novel agents, active templates. The structural diversity and biological activity of natural products make them the most valuable sources of drugs and drugs lead. In fact, more than 1/3 of the US FDA-approved therapeutic agents over the past 20 years are derived or inspired by natural products. So most of it is actually, for example, this NM, they are inspired from natural product, okay, or natural product derivatives, okay. And this is natural product biologics, and this is a purely natural products, okay. So we can get this information in Newman and Craig in 2020, which is an updated review on natural products as sources for new drugs over nearly 4 decades from 1981 to 2019. Okay? Right, in the next table next to the charts are examples of representative drugs discovered from natural products taken from Lee et al. In 2019. The list actually include several well-known history-changing drugs such as penicillin, morphine, paclitaxel, and artemisinin. I know artemisinin is actually making a big, headway, but of my particular interest is actually andrographolites. Okay? So andrographolites, sorry. Right. So andrographolites actually has been found, can be found in abundance in our fauna. Okay? In both our faculty in UNED and USM, this is a very active research project, and I know for a fact that the Dean of Facultas of Pharmacy in Universitas Airlangga actually work on andrographolites as well. And in fact, this is actually one of my early research work back in 2003. Okay? So I work on molecular docking of andrographolite to rennin, HIV protease, and tyrosine enzymes back in 2003. What I'm trying to say is like we in both countries actually work a lot on these natural products, and it's yet to see whether we can hit the FDA market, the market worldwide. But I am glad to know that in the case of andrographolite, this compound just recently completed various clinical trials in United States and Europe on ulcerative colitis. So let's leave andrographolite for now and let's turn our attention to selected milestones of drug delivery from natural products. Go to the next slide. Okay. All right. The first, this is just a brief timeline of selected milestones of drug discovery from natural products. Human probably already aware of medicinal properties of various plants since more than 6,000 years ago. As a Muslim, I believe the first human being, Prophet Adam, already knew this, wallahu alam. But in ancient time, plant-based drugs rich in bioactive natural products were used in the forms of oils, infusions, and pills to treat colds, coughs, and other diseases. The oldest recorded text for the use of natural products as a therapeutic agent was in Mesopotamia in 2600 BC. Okay? There are several milestones throughout the history of natural product-based drug delivery. The first one actually is when morphine was first isolated in 1803 from opium seed, and in 1827 this opiate was commercialized, okay, making it the first pure natural derived medicine available in the clinic. Right, this spurs the discovery of some well-known natural products, natural compounds or major herbal preparations, for example, quinine, colchicine, codeine, salicin, papaverine, etc. And then Fleming discovered penicillin. Okay, so this happened in 1929, where people think that this is the golden age of natural product-based drug discovery. Okay, so it's the discovery of penicillin really revolutionized drug discovery research, and in the year 1952. Ushered the discovery of streptomycin by Salmon A. Waxman. Okay? Right, and this results in Nobel Prize in Physiology of Medicine accorded to Fleming in 1945 and also to Waxman's in 1952. Okay? Since cytarabine becomes the first FDA-approved marine derived anti-cancer drug in 1969, exploitation of marine environments especially has led to encouraging outcomes. So far, more than 25,000 marine natural products have been identified at a rate of 1,000 new compounds per year since 2008, including approximately 1,490 new compounds in 2007. So this shows the opportunity that we can work on natural product even on our marine. And as a countries like Indonesia, which you actually have, you're surrounded by waters, right? So this is a great opportunity, okay? Right. So many of these marine natural products have been applied in medicine and agrochemistry and commercialized, such as cytarabine, vidarabine, eribulin, zicotonide. Okay. Right. So the history has demonstrated that natural products are great source for drug discovery. In 1990s, pharmaceutical companies scaled back or abandoned their natural products program and focused on combinatorial chemistry and high throughput screening to generate drug candidates. Okay. Unfortunately, this strategic shift correlated with the overall reductions of novel lead compounds in the drug development pipeline and substantial decline in new drug approval. For natural product community and champions, the climax of the story emerged in 2015 when the Nobel Prize in Physiology or Medicine was awarded to Campbell, Omura, and Yu Tu for the discovery of ivermectin and artemisinin. Right, so this is actually the highlight for natural product research, because artemisinin not only is a traditional medicine coming from Artemisia annua, but this is a drug that has significantly reduced the mortality rates of patients suffering from malaria, and we all know that we are still affected by malaria, and now malaria is actually demonstrating some resistance towards the drug. So the discovery of artemisinin is actually very crucial to the world, actually. Okay. I mentioned earlier on that in 1990s, pharmaceutical companies scaled back or abandoned their natural products programs and focused on combinatorial chemistry and high throughput screening to generate new drug candidates. But they don't really have a good success rate, right? But the question is, why do they abandon natural products program in their discovery? All right. So the challenges then for natural product drug discovery is actually in the olden days that, or even now, most efforts of natural products were put in top-down approach. Actually, we initiate by screening biological samples of desirable bioactivities, followed by the compounds isolation and characterization. Okay, and the results from large screening collection will suggest the diversity within biologically relevant chemical space. Okay, so but really the challenges of a natural product is actually when we actually working on it, we try to isolate the compounds. Most of the time we actually discover known compounds. So in terms of patentability or commercialability of that compounds, not very attractive for pharmaceutical company. Right? And there are a lot of technical barriers of the isolation of compounds from extracts, and again, why, despite that we work on endocrofolite, for example, for many years, or tongkat rali for many years, we are yet to produce our own drugs from our own soil to the world. Okay? But we're stepping back, because I was very interested in the talk by Ibu Maya just now. So there is hopes for the natural product people. So hopefully that Malaysia will catch up with Indonesia in that respect. Okay. And also natural products are actually quite incompatible to high throughput screening platforms that the pharmaceutical industries have actually invested on. Right. But there is still hope for us. Okay. The advent of novel technologies, including innovative extraction of natural compounds, computer-aided drug design, and various omic technologies can create potential for discovery of novel drug candidates either as natural products or inspired or modified natural products. Okay? Innovative extraction technologies, including semi-bionic extraction, supercritical fluid extraction, microwave-assisted, ultrasonic assisted, and enzyme-assisted extraction. All of these can actually be used to extract natural compounds efficiently from plants. Okay? So these extraction strategies have been shown to have similar success to the traditional methods, allowing extraction process to get most compounds from the natural products. Okay? Then we also have a genomic-based techniques that represent an effective platform for natural products identification, but different parts of the same plant with similar sequence may have different qualities. And we all know that plants, grown in different even environments also will give a different bioactivities. Okay, so we can use technology like DNA barcoding to actually identify plants. Okay, so that they actually have similar sequence and almost the same quality and clinical utility, right? But also other omics technologies such as the genomics, transcriptomics, proteomics, metabolomics, and metabonomics. But my talk today will focus on computer-aided drug design. Oops, right. Right, so computer-aided drug design, because recent advances in computer-aided drug design techniques actually provide powerful tools for processing complex natural products and developing novel drugs based on their structures. Okay, so such techniques actually include machine learning, simulation, whether molecular dynamics simulation or molecular docking simulations, cheminformatics, and such bioinformatics, as well as structure-based drug design. So in this talk, I would like to share some of our natural product work aided by computational techniques. Okay, so why we want to work on natural products and apply it with the computational techniques? As a general background, actually, I am a pharmacist specializing in pharmaceutical technologies. My PhD is actually on applying molecular dynamic simulation for pharmaceutical problems. So I thought that because Malaysia is actually have mega biodiversity, right? So maybe we could apply computational techniques to actually expedite our research. Okay, so just like Indonesia, which is more mega biodiverse than us, right? Malaysia, our forests actually contain about 15,500 species of flowering plants. Okay, and we have all these other millions of microbes and invertebrates, which can also be sources of potential drugs. Okay, so this information actually provide us the motivation to work on the natural product, but not really natural product per se, because I'm not a natural product scientist and I do not want to claim that I am a natural product, but actually with the utilization of computer-aided drug design to expedite the natural product research. All right. Since we understood the values of our mega biodiversity and how important to gather as much information on them, we developed a database system which can contribute to drug discovery back in 2005. Okay? So our database is slightly different from others in the world because we actually put as much information as possible on the plants and their use as traditional medicines, as well as on the compounds that we isolated from them, so that we can use these compounds to screen to find new scaffold or new drugs, okay, new potential drugs. This database we call NADI, so it's actually a one-step solution for natural product. This is what we envision. Whether it's actually a reality or not, this remains for the people who actually use it. Okay, right. So NADI can be actually achieved at http://www.nadidiscovery.com. Okay. And you can search the compounds, you can search for the plants, you can search for the compounds, you can do substructure search and structure search as well, using this thing. This is how you can use it. I didn't want to show it live because something might just happen wrong. But there are about 400 plants which give about 4,000 compounds in this database. Okay, you can just search. For example, you are interested in quercetin, so you just type quercetin and you can get all the different the quercetin, the actual quercetin, and also the derivative of quercetin. This is like Google search, okay? Right, you can do some substructure search, okay, or structure search, right? But while we're developing this, we actually have a specific things in our mind. We want to have these compounds used for virtual screening, and mostly for our case, we want to have a complete control what we can do to answer all the questions that we want. So for example, we have questions whether we can identify plants for further extraction or isolation, or whether or not we can use the active natural product that are virtually found to design simpler and more active molecules, or because we are very interested in the traditional use and we want to correlate it between the ethno or traditional use with the scientific explanation. And we can we identify types of compounds against a specific disease targets. So those are our major questions. Okay, right. So basically, with this thing, we use molecular docking, especially with for virtual screening. Like yesterday, I gave a talk on the molecular docking in drug discovery. So what we do in molecular docking is actually to identify ligands which has a binding mode, because this is a binding mode will tell whether the ligand will be happily sitting in the active sites of a protein target. Okay, and then we want to predict the binding affinity. Okay, and the results of this docking, we actually use it for rational drug designs. Okay, so we can use to find inhibitors for specific proteins and design new drugs. Okay. All right. It's a very short clips. Maybe it's not even 1 minute, right? So show how molecular dockings looks like. So this is just like say your natural products, potential drugs which going to bind to a protein, and we are looking at what is the interactions between these drugs and the proteins. Just remember that proteins are large biomolecules that are made up of amino acids. So these are 20 natural amino acids. This string of amino acid actually fold in a particular way that determine the shape of the proteins. And if you look at the protein structure properly, you will see that motifs such as the secondary structures such as helices and sheets and loops. Okay, all right. So with this information on the structure of the protein and the structure of the drug, we can see how they actually bind. So typically these are the type of interactions that happens during docking. We will look at whether hydrogen bond is determining the strength of the binding or hydrophobic interactions of the molecules with a protein, or also the how close and the strength of these hydrogen bonds that determine the binding or the different types of interactions such as pi-pi interactions with the protein. So all these we use in the analysis of our docking results. Okay. All right. So proteins, so ligands that fits nicely to the binding site and try to avoid the binding, the clashes or straight conflict with the protein. These are the ligand that we want to really understand and see how we can improve the molecule further to make them as a potential drug candidate. Okay? Right, so this is another way of looking at it. So this is how the binding site, where just now I showed how the ligand binds. Okay, so small drug molecule can be found and designed to feel the binding site. So the more contacts that you make, the more stable the molecule will be. Okay, right. So with a little bit of introductions of the docking, we will use it to actually find the particular compounds which have a high affinity towards a specific disease targets of interest. Okay, right. So you can use molecular docking either as a general virtue screening, where you have many compounds where you dock in one particular drug targets. This you want to find the best compound for the binding into that protein. But in the case of I just showed an example of natural product. You probably want to dock all this natural product in the system, for example, right? So, but we also have a different types of docking where you, for example, one day you found an unknown compound, right, of unknown activities, and you have no idea, for example, what it would be used. So you can use a reverse docking technique where you can see whether this ligand will act on any particular proteins. You can dock in many proteins to see what is the best target for this particular compound. All right. So let me just demonstrate how we use it in our work. So in 2015, we reported our discovery of plants with medicinal properties which have anti-neuraminidase activity. Okay, so the work involved virtual screening of our nature product database, again neuraminidase from H5N1, right? So I just want to show here that we have Ibu Neni Puritasawi, who actually did his master's at that time with Professor Nonisa Muhammad, right? So this is actually a lot of master students' work and one PhD student, Dr. Mokhtaridi, to come up with the workflow of a work. What we do here is actually to actually give a confidence of whether our work is giving the right predictions. We actually validate it with the known inhibitors, okay? Right, so and then when we are very confident with our methods and our parameters for docking, so we actually apply it using our database compound from our database to screen the NADI to find a potential neuraminidase inhibitors. Okay, so the receptor we choose, we took it from protein data banks. Okay, and from the 4,000s that we actually, I think not even 4,000 at that time, it was 3,000 plus compounds. Right? So we actually ran the 100 active compounds and grouped them according to the plants, and we tested the plants with the top predicted ligands. And these plants we actually work further for extraction, fractionation, and isolations. And then we actually determine the bioactivity of the plants, okay, and the compounds, okay. All right. So the 5 plants that possess the greatest number of predicted influenza inhibitors are actually Manggis, okay, Garcinia mangostana, Momorita charantia, Tidivertica, Busia javanica, and Euchroma longophilia. Okay, so we actually tested on the sorry, I just want to get to my pen. Okay, we just tested on the doesn't work. So we just tested on the H5N1 neuraminidase, and this plant gave actually quite good activity, except for Tongkat Ali. Okay, right. So this is a nice collaboration with UNER. Okay, at that time, Ibu Nani was joining us, and we were quite pleasantly surprised to see now my what is it? Hold on. My computer froze. Okay, what am I going to do? But anyway, I can move, and then when it's defroze, we will go back. Okay. So what we did is actually, what we found actually 12 compounds that we isolated from the plants, these 5 plants, and some of the compounds actually was not present during our screening, but it appeared, and it actually also give a good inhibition activity on the neuraminidase. Okay, I tried to work. Maybe I should. My computer froze. Can you see the other side? No. Right. Let me just stop share. Okay. Let me just stop share so I can. I'm so sorry. My computer froze. So these are the thing that when things goes wrong is gonna go wrong. Right. Can I just stop, Ibu? You think it will be okay?
Yes, go ahead.
Yeah, I just go on. Okay.
Five more minutes, Professor.
Okay, but it's very interesting that this strategy is actually able to identify plants that possess the greatest number of H5N1 inhibitors rather than a single H5N1 inhibitor using docking screening, which shortens the sample preparation time for the primary validation. Okay, so what do we believe that the protocol is not, we believe this is actually written from another group which cited our work. They believe that the protocol will significantly facilitate the practical application of virtual screening of natural products. So this is the first time we have a good workflow which actually can expedite the natural products discovery. So this is cited in phytomedicine by a group in China, right? Yeah. So we actually use NADI for a lot of other work. I'm so sorry that you cannot see it now. I don't understand when it froze. Right. Did I do something with a pen? Right. Okay. Anyway, but anyway, we have a lot of work on neuraminidase. That's not our publications. We follow on the work with H1N1, where we design a new compound based on ferulic acid. Well, we found that during our screening with H1N1, we actually isolated ferulic acid, which have a structure similar to cinnamic acid. Is a derivative of cinnamic acid, but the structure similar to curcumin. Okay, so this ferulic acid has shown to have antioxidant, anti-cancer, anti-aging effects. So we thought that maybe ferulic acid can be used to design new compounds for neuraminidase, right? So we actually so unfortunate that you won't be able to see the slides. Okay, ferulic acid does have some similarity with the drug oseltamivir, right? So the structure has a functional group, 3 functional group, carboxylate hydroxy methoxy group, which probably contribute to the interaction with neuraminidase influenza neuraminidase. So we were able to design new compound. This is done by Pak Mewan Haryono, who obtained his PhD. So he actually managed to design a compound with a very good activity, about 50 micromolar activity. Okay, we tested from the enzyme inhibition assay. We tested on antiviral assay, and sure enough, this compound show a very good selectivity and safety index, in fact, better than oseltamivir. Okay, so the compound also shows a good EC50 to the virus. Okay?
Professor, your time is up.
Okay. All right. So we have a patent on this as well. Okay, and we also apply it to dengue and a lot of other work. But I have a very nice slide that I want to show about COVID-19, because when in 2019 and 2005, when I started the NADI database, we have H5N1 pandemic. And then in 2009, we have H1N1 pandemic. Then Dr. Nani joined USM as a master student. Ibu Nani joined USM as a master student, and we collaborated with ONER on H5N1 and H1N1 research. Okay, so I have a very nice slide to and tease UNER whether maybe we should take our collaboration to the next level by working on natural products for big data discovery for the treatment of COVID-19. Okay. So with that, I would like to acknowledge all my collaborators, especially Professor Osada in Weekend, Dr. Romy Amaro in United States, California, San Diego, my dear colleagues Izatul, Dr. Rosa, and our collaborators in Prof. Nusada, Dr. Rosana Kalija, and all my former PhD and master students. Okay, so thank you very much. I would love to answer a few questions. My screen froze, so I will not be able to see the
Question you mean?
the question.
Yes,
But
I will read it for you.
Okay.
Yeah. Thank you very much, Professor Habibah, for a very interesting presentation. Some highlights from Professor Habibah's presentation is that there will be no more difficulties associated with isolation of known compounds because of the fast development of technologies. And in this context, computer-aided drug design comprises a broad range of theoretical and computational approaches. Those are part of modern drug discovery. So CADD method nowadays becomes the key contributors to the development of drugs that are in clinical use or in clinical trials. Such methods have emerged and evolved along with experimental approaches used in drug design. So Professor Habibah, both of us have huge mega biodiversity. So collaboration is needed for the application of traditional medicines, traditional use with scientific explanation. Is that correct, Professor Habibah? So we will proceed to discussion later at the end of this session. Thank you very much again.
I can try to fix my computer. Thank you very much, Ibu. Thank you.
Okay, distinguished guests, ladies and gentlemen, now is the time for the third speaker. Associate Professor Dr. Konkanok Inkaninan. Here is her short profile. Dr. Inkaninan pursued her PhD degree from Division of Pharmacognosy, Leiden University, the Netherlands. Her research interests are research and development of natural health products, and she is very active in cannabis research for medical purposes. This is very interesting. Her experiences on technology transfer will include Brahmi food supplement, hair growth promotion products containing curcuma aeruginosa extract, anti-cellulite herbal compress. And she is the adjunct professor of the Faculty of Pharmacy, Airlangga University. She received many awards, such as Brahmi Award, and this is very interesting. Today's presentation of Dr. Inkaninan is entitled Hemp, a promising herb for food, drug, and cosmetics. Dr. Inkaninan, sawadika.
สวัสดีค่ะ Selamat pagi.
Selamat pagi. We have not met each other, but we are together.
Yes.
So
amazing. COVID-19 to us.
So,
the technology, online technology.
Yeah. So hopefully that you are able to visit Surabaya soon.
I hope so.
So Dr. Inkaninan, you have 30 minutes, and I will give you a reminder five minutes before the due time. Right. So please welcome Dr. Imkaninan.
Hi.
Time is yours.
You have my video clip, right?
Yes.
Because I'm afraid that the connection would be a problem, so I send my presentation through the video clip, and then I will be here for discussion and answering all of your questions. Thank you.
Okay, great.
Good morning and selamat pagi. First of all, I would like to thank the organizing committee and Airlangga University for the effort and determination to make this online meeting possible. The topic of my talk today related to the very hot issue in Thailand before COVID-19 came, which is the use of cannabis. And today I would like to focus on one strain of cannabis, which is hemp. The outlines of my talk today include introduction, and then I will talk about two kinds of cannabis, which is marijuana and hemp in Thailand, our regulation and marketing opportunities, and then chemistry and biological activities of hemp, health benefits and concern, and then finally I will talk about research on hemp at Naresuan University. The recent taxonomist community conclude that all cannabis share the same scientific name, which is Cannabis sativa. It belongs to Cannabaceae family and it has several names such as cannabis, marijuana, gansha, and hemp. The best way to define hemp or industrial hemp and marijuana is by the level of THC, the measure of cannabinoid that can get you high. Hemp has low level of THC, which is controlled by the law of each country, while marijuana has high level of THC. In general, the morphological differences of hemp and marijuana are that hemp is taller, the leaves are narrower when compared to that of marijuana. Some call hemp as Cannabis sativa subspecies sativa and marijuana as Cannabis sativa subspecies indica. However, several strains of cannabis has been developed and many hybrids with mixed morphological characteristics are found. Hemp is a strain of Cannabis sativa species that is cultivated for industrial uses for its fiber, sheath, seeds, and other products. Hemp is legal in many countries for industrial uses, while marijuana is only legal in limited countries for medical uses and recreation. It is known that both of hemp and marijuana are listed as narcotic drugs in Thailand. In European countries and North American countries, to be legally classified as hemp, the crop may not contain more than 0.2% or 0.3% of THC respectively. In Thailand, hemp may not contain more than 1% of THC. In Thailand, cannabis known by the name of gansha is classified into class 5 narcotic. Recently, Thailand has launched a new medical marijuana law that allows the medical use of cannabis. This new law states that the use or possession of cannabis for the purpose of medical treatment of patients or for research is allowed. For the treatment, cannabis has to be prescribed by the medical professionals, including folk doctors. The first legal crop of cannabis in Thailand was produced by Governmental Pharmaceutical Organization or GPO last year. They made the extracts containing high CBD, high THC, and CBD to THC 1:1 under Special Access Scheme (SAS). There is also a movement on regulation of hemp in Thailand. The new law in Thailand allows hemp to contain no more than 1% of THC by weight in its dried flowers and leaf. Hemp is one of those miracle plants that every part can be used. Flowers can be used in drug and health product industry as it contains CBD with low level of THC. Hemp seed and hemp seed oil are food and animal feed. In addition, oils can be used in cosmetic and healthcare products. Fibers can be used in pulp and paper, insulation material, textile, and automotive industries. Sheaf from stalk can be used as bedding material for animals and in construction industries. Leaf can be used in health products industries. This makes hemp as the promising industrial crop. In order to promote Thai hemp industries, in August 2019, CBD, hemp extracts, seed, and seed oil have been removed from the list of narcotic drugs. The parts of hemp that are now legalized are bark, stem, fibers, and their products. Hemp seed and hemp seed oil used in food and cosmetic preparations. CBD extract from hemp with THC not exceeding 0.01%. Hemp extract that contain THC not exceeding 0.2% by weight can be used in drug and herbal products. Only local licensed producers will be allowed to use parts of the plant in their products within 5 years. This is the important message. Marijuana and hemp are still in class 5 narcotics. The cultivation, import, export, sale, possession, and consumption of marijuana and hemp without permits are still illegal. At this moment, the one and only hemp grower in Thailand is the Highland Research and Development Institute. Their hemp strains are known for their high-quality fibers, which make them suitable for using in textile industry. With the new law expecting to be launched this year, there is possibility that the private sectors will get permission to grow hemp. This slide shows the content of THC and CBD info strains of hemp grown by the Highland Research and Development Institute. You can see that THC of these 4 strains is not exceeding 0.3%, but CBD is not that high, it's only 0.5% by dry weight. Next, I would like to talk about chemistry and biological activities of hemp. The active components in cannabis are cannabinoids. The major compounds are THC or delta-9 tetrahydrocannabinol and CBD or cannabidiol. THC is psychoactive. It is CB1 and CB2 receptor agonist. It has many effects to the body, including euphoria and relaxation, sleep and drowsiness, appetite stimulant, painkiller, anti-emetic, and muscle relaxant. However, in hemp, THC is limited as less than 1%. This makes another cannabinoid, CBD, is a major component in hemp. It is CB1 receptor antagonist. The biological activities of CBD are anti-anxiety, neuroprotective, anti-convulsant, antipsychotic, anti-tumor, painkiller, and anti-inflammatory effect. THC has been approved for the treatment of chronic pain, multiple sclerosis, anorexia, and chemotherapy-induced nausea and vomiting. while CBD is used for reducing pain and inflammation and controlling epileptic seizure in patients with childhood epilepsy, Dravet syndrome. Cannabinoids and monoterpenes are produced in granular trichomes, which are found mainly on pistillate of female flowers and some in leaves. This is why the level of cannabinoids is high in pistillate flowers, moderate in leaves, and low in stems and roots of cannabis and hemp. How cannabinoids affect the body? There are two types of cannabinoid receptors in our body, which is type 1 or CB1 and type 2 or CB2. CB1 found on nerve cell in brain, frontal cortex, the limbic system, hippocampus, hypothalamus, pons and medulla, spinal cord, and peripheral nervous system. Low level in salivary glands, leukocytes, spleen, heart, urinary and GI tracts. CB2 highly found in immune tissue and immune cells, spleen, tonsils, and thymus glands, monocytes, macrophage, beta cell, and T cell. Not only THC and CBD, but there are also more than 750 substances in cannabis. This includes cannabinoids, both in acid and neutral forms, terpenes, and flavonoids. Cannabinoid presents in plant in acid form such as CBGA, THCA, CBDA, and CBCA. It will change to neutral form such as CBG, CBD, and THC, with some interventions such as heat and post-harvesting process. Among these cannabinoids, THC, CBN, and delta. In hemp, THC and their analogs is low, and the major compound is CBD. Not only THC and CBD, other cannabinoids in hemp also have some biological activities. For example, CBG, CBC, CBN. Another group of compound monoterpenes give characteristic aroma in cannabis. In addition, they have some therapeutic effects such as antimicrobial, anti-inflammatory, and enhance the cell penetration of other compounds. Many cannabis users and some scientists believe that the cannabis extract is more effective than the one isolate compound like THC and CBD, as many compounds will give synergistic effect, which is called entourage effect. Anyway, I would like to share some results in vivo experiment of CBD. CBD has anti-inflammatory effect. Transdermal cannabidiol reduces inflammation and pain-related behavior in rat model of arthritis. And another study showed that CBD has a potent arthritic effect in murine collagen-induced arthritis through its combined immunosuppressive and anti-inflammatory actions. The clinical studies of CBD show the positive results in the treatment of epileptic patients and also children with Dravet syndrome. This slide shows physiochemical properties and pharmacokinetics of CBD. CBD is highly lipophilic. Therefore, the oral bioavailability of CBD is very low, while its bioavailability via inhalation is higher. The elimination half-life of CBD is quite long, 18 to 32 hours. Cannabidiol is metabolized in the liver as well as in intestines by cytochrome P450 enzymes. In 2018, CBD was approved by FDA under the trade name of Epidiolex for the treatment of two forms of the treatment-resistant epilepsy, Dravet syndrome and LGS in children. Even though there are a lot of CBD products in the market, under U.S. federal law, CBD is still illegal to sell as food, dietary supplement, and animal feeds. In 2019, the European Commission announced that CBD and other cannabinoids would be classified as novel foods. In some countries, food beverage items can be infused with CBD as herbal additives. However, Thai FDA still do not agree with this. In the markets, there are hemp extracts, which sometimes called hemp oils, that's extracted from female flowers of hemp. Sometimes it's called full spectrum hemp extract, and hemp oil containing cannabinoids, monoterpene, and other compounds, and is claimed to have antiretroviral effects and give many health benefits. These are examples of health products containing CBD in the online market. For example, CBD cream for pain relief, CBD oil as dietary supplement, and CBD in water, in cookies, in gummy bear, in cosmetics. However, there are concerns of FDA about CBD products. First, there might be potential liver injury caused by CBD, and CBD might interact with other drugs. For example, CBD interfere with cytochrome P450, a group of enzymes that are important to drug metabolism. CBD might show male reproductive toxicity. It gives drowsiness, and the potential effects of sustained or cumulative use of CBD and co-administration of CBD with other medicines, or the risk of vulnerable populations like children, pregnant and lactating women, and elderly, unborn children, and certain animal populations. In addition, transdermal penetration and pharmacokinetics of CBD still unclear. The effect of different routes of CBD administration, such as oral, topical, inhalation, on its safety profile must be studied. Next, I would like to talk about something that is more safe in hemp, which is hemp seed and hemp seed oil. Hemp seed is important source of nutrition. Hemp seed contain more than 30% of oil and about 25% protein with dietary fiber, vitamin and minerals. Hemp seed oil made by cold compressed. It contains more than 80% of polysaturated fatty acid or PUFA with linoleic acid, which is omega-6, and alpha-linolenic acid, which is omega-3. The ratio of omega-6 and omega-3 in hemp seed oil is 2:1 and 3:1, which is considered to be optimal for human health. The two main proteins in hemp seed are adenosine and albumin. Both are easily digested and contain nutritionally significant amount of all essential amino acids. Hemp seed can be used in food, salad, cookies, and hemp seed oil can be ingredient in cosmetics. Because of these PUFA and other fatty acids, give some benefit to skin. However, cannabinoids is considered by FDA as contamination and shouldn't be found in cosmetic product containing hemp seed oil. Hemp seed can be used in animal feed as well. Next, I would like to share you some of my very beginning results of research and development of hemp for medical uses at Naresuan University. We have the project funded by ARDA to study from the growing of hemp, the strain development, and the used and extraction, chemical components analysis, toxicological study, pharmacokinetic studies, pre-formulation, formulation study, and pharmacological studies. And the aim of this project is to develop some preparation containing hemp extract. If you want to do research with hemp, one of the very first things that you have to do is ask for permission from FDA. So we have the license to grow hemp, to have hemp in our lab, and to make hemp extract. And for security reasons, we have to prepare our laboratories and show this to Thai FDA to allow us to have hemp and hemp extract in our lab. Comparing to cannabis samples, in cannabis samples that we have, there are high THC and very low CBD. This is the samples we have from the police acid cannabis samples. But for hemp extract, there is high level of CBDA and CBD and low level of THC. And with the analysis of headspace, GCMS, we could analyze monoterpenes, sesquiterpene, and cannabinoids in hemp and in cannabis. And you can see that this is the sample of cannabis. There are more cannabinoids compared with hemp, which only CBD can be detected. Last but not least, I would like to thank funding agencies, ARDA, PERS6, T-Cells, to fund us some research grant to do research on cannabis and hemp. I would like to give sincere thanks to the Highland Research and Development Institute to provide us hemp materials. And I would like to thank Naresuan University for all the support. Thank you very much for your attention. Any questions are very welcome.
Thank you very much, Dr. Inkandinan. Talking about cannabis, always very interesting for pharmacists and other health professionals. So, some highlights. Hemp has been developed for therapeutic use and other consumer products. It contains cannabidiol, which is commonly used for epilepsy. It has also showed potential health benefits to manage in preclinical model central nervous system disease such as epilepsy, neurodegenerative diseases, schizophrenia, multiple sclerosis, and anorexia. CBD also showed anti-anxiety, anti-nausea, anti-arthritic, antipsychotic, anti-inflammatory, and it has immunomodulatory properties in vitro and animal studies. So recent clinical studies have indicated hemp seed oil can be used as promising functional food, animal feeding, and cosmetic ingredients. Dr. Inkandinan is Associate Professor in Center of Excellence in Cannabis Research and Bioscreening Unit, Department of Pharmaceutical Chemistry and Pharmacognosy, Faculty of Pharmaceutical Sciences, Naresuan University, Thailand. Dr. Inkandinan, can I buy CBD cream
Right now, right?
Yes, I want to go to your place.
Right now, actually, I mean, it's the serious question. Right
No,
now,
no.
if you um, when you want to buy CBD cream, it means that we have to register it first. So in Thailand, if it's CBD cream, and if we prove that it has lower THC, I mean the amount of THC lower than 0.2, then it's not narcotic drug.
I
It means that
see.
we can register, but depends if we want to register as a drug, a topical drug, or we have another category called herbal product. So now we have the new department called of FDA called herbal product. So the Thai, because Thai government try to encourage industry and also researchers to do research on hemp, so they kind of open the opportunity to register CBD at the as a herbal product. So right now it's still not possible, but maybe next year is possible.
Okay, I will come to your place next year, for
Yes,
sure.
please. Now
So-
we have a clinical study of CBD cream for psoriasis. Actually, it's ointment. It's
Ointment for psoriasis.
for size study with 4 hospitals.
Oh, great.
On psoriasis. Yes.
Thank you, thank you. So, dear audience, now it's the time for a discussion. The scientific committee has already selected some questions from the audience, so I'm going to read it one by one so that each speaker can give direct answers. So, question goes to Professor Habibah. The question is, results by HTS often promise a natural product, especially plants, can become a new drug. But in fact, when the material is continued in vivo testing in animal models, the results are not always satisfactory until some are declared inactive and the research is stopped. How do we solve this problem? Professor Habibah, are you there?
Hello.
Professor? Yes.
All right. So from what I understand, this is actually a very general questions. Sorry, I changed my background. So these are the slides that
Later on, you have to tell us about your COVID-19 research.
Okay, no, sorry. So you're right. We actually do whatever we can do before the testing goes to human. So you're right. You said that once we do the in vitro first. This is not only for natural product, for any other potential drug candidates, right? We will test it in vitro, and then we go to in vivo on animal before we go to clinical. And it's interesting that sometimes we fail at the animal study. So what do? We have to go back to the drawing board, to understand why the molecule doesn't exert the action that it's supposed to have. Okay, but I wonder whether you know that even if it's active in the animal study, doesn't mean that it is active in human, because we actually, although similar, but we are not very similar to the animal, right? So all these things must be taken into account. So there is no standard answers. You really have to go back to the drawing board, to really understand, the physical chemical characteristic of the chemicals that you actually design, and where can it be wrong? I want to highlight one thing which I remember clearly is like when they try to discover zanamivir, the first anti-flu drug. So what they do is they actually attach the guanidinium, so they become you always use Dana, hear about Dana. So Dana is active in vitro, and it's actually not active in vivo.
In vivo.
So they have to go back the drawing board and then try to improve the drug, whether to improve the lipophilicity, solubility, and whatnot. So it's not a very straightforward answer to give. It can fail in any part, even it can fail in clinical trial, right? But the thing is about drug discovery is actually they have this concept that if you want to fail, you have to fail early because it's going to cost a lot of money when you fail towards the end. Okay, I hope I answer the question.
Thank you, Professor. So, question for Dr. Konkanok. This is a question from Professor Suharyono from Faculty of Pharmacy, Universitas Airlangga. In Indonesia, hemp or marijuana is still pro and cons for medical use. But I remember in the past time, before 1950, extract cannabis spisum ever used by prescription for sedative and hypnotic. But in Indonesia, marijuana and its product were banned for use because narcotic is very dangerous of the abuse because of it's abusive. Now in online, many marijuana product like CBD oil is on sale for many therapeutic for many diseases. So his question is there a marijuana plant in Thailand or maybe hydroponic for plant? That is his question for Dr. Konkanok.
Can you repeat the question again?
The question
the growing
is,
of cannabis.
yeah, is there a marijuana plant growing in Thailand or we have to use hydroponic technique for plantation?
Oh, okay. So right now there, first of all, the marijuana and hemp are still in the category of narcotic 5. So people have to get a license to grow it, and the private sector cannot grow it alone. I mean, by their own, they have to collaborate with governmental organization.
Okay.
And then there are many. Right now, there are, I think, at least maybe 5 or 6 or even more license from the university and research institute with collaboration with private sector to grow hemp with different techniques. Hydroponic is one of the technique that they use, but because of the cost of growing is very high, so they right now people are trying to have the more effective but cheaper techniques, so they are thinking about that. But the problem of hemp and cannabis is it attract insects and fungi. So if we grow it outdoor, I mean, like underground people, like illegal cannabis, they use a lot of pesticide and fungicide, which is not good for medical use. So we have to study how to grow it in a cheap way and prevent and the way that it's clean and it's in medical grade. But hydroponic is one of the technique, yes.
Okay. There is a question, Dr. Konkanok, from Mahardian Rahmadi from the Faculty of Pharmacy, Universitas Airlangga. This question is, instead of analgesic and anti-inflammatory effect, how is the addiction potential of hemp? Because 1% of THC is quite high concentration to induce euphoria.
The addiction, right? You are asking about the addiction.
Addiction.
I have no kind of scientific answer yet, but from the review, they compare tobacco and cannabis, and then they say that the chance to get addicted by cannabis is less than tobacco. But of course, the side effect of cannabis may be high because of the the psychoactive effect. So that's my answer. That it can be addicted, but it's not, as have higher chance than tobacco. It's just the same.
I see. I see.
Mhm.
Okay. Okay. Professor Habibah, you have a question. You have a question related to COVID-19 from Dini. What about COVID-19? Do your research team also conduct research of drug discovery on COVID-19? That is her question.
You know, at the beginning of the year, when we started having COVID, we actually do a lot of work on, virtual screening of COVID-19. Even we do repurposing, drug repurposing. But my experience as an editor of journal, those virtual screening, if you don't validate it with some experiment, will not be able to land you any good publication. So we actually stop it, but the results is there. We're waiting if we can validate whether any of the drug targets become available that we can buy, and then we can do the in vitro experiment, or we can find a good collaborators to test our compounds, on the molecules. Otherwise, it's very difficult to move. If you just use work on the computer, it doesn't go anywhere actually. Right? We want to actually find cure, so we want to find a good experimental validation system. I hope I answered questions.
Okay. By the way, Professor Habibah, are you familiar with galangal rhizomes, Kampferia galangal, cekur in Malaysia?
Yes. Yeah.
It's very, very popular in Indonesia for respiratory tract infection.
Maybe we should do something about it. We should look. Ibu Prof, you know that andragofolite is actually in the list of for treating COVID-19 in China. So yeah, we will see the results of any trial over there. So there are a lot of opportunity, but the thing is, if we can test it, because the virus is very dangerous, nobody wants to work on the live virus at the moment. Yeah. We also do not want to expose ourselves. Yeah.
All right, then. Another question, also for Professor Habibah. Is NADI database for Malaysian plants only, or is it for general?
All right. So the plants is actually available. The plants are only 400. So I believe these plants also can be found in anywhere in Southeast Asia, especially Indonesia. But Ibu, we actually have a NADI Indonesia. It's actually hosted in Pajajaran University, because I give it to Pak Mukhtaridi. To I forgot about it. To actually, you can use. It's open to use.
Okay. Thank you. Professor Dr. Kongkanok, you have another question. The question is, based on your explanation, what is the morphological difference between hemp and marijuana? Are they the same, or that is the question?
Okay. Generally, hemp is taller, like very tall, and then the leaf is more narrow.
I see.
So kind of marijuana is smaller, and the leaf is broader. But right now there are so many strains of hemp and cannabis, and sometimes they mix everything. So you can find you can easily find some strains of cannabis that is tall or some strain of hemp that is small. So, but generally it's just like that. For hemp, there's a lot of fiber. So people grow it and need fiber. So they make the tree tall. So they grow hemp together so that the tree going taller and yeah, with narrow leaf. Yes, a lot of fibers.
Okay. Dr. Konkano, have you ever heard about north part of Sumatra Island of Indonesia, that cannabis grows very easily, good quality of cannabis?
I believe so. Yes.
So there should be a collaboration between
Right.
us.
I think between government first.
Yeah,
Has to be
yeah.
the policy of the government.
I
Yes.
see.
Yeah.
It's still it's illegal
Yeah.
in Indonesia.
It's I mean, it need a license. I think in Indonesia, I think it's possible to do research as well. But then for the license, you have to think about how to get a license. I'm quite sure that there is a way for research to do. I mean, to do research on cannabis. And if you discuss with FDA, and there might be channel to go, right?
In
With
your
the
country, who gives the license? Ministry of
Sorry?
Health? Who gives the license in your country?
FDA.
Oh, FDA, I
Ministry of
see.
Health. So before, I think 3 years ago, before the new government came, if we want to do research on hemp on cannabis, it's possible. We can ask for like research reason, and then we get the license, but we cannot use in human. So because it's not allowed by law. So last year they changed the law that people can, it's legal to give cannabis to people for medical benefit, but all of us need a license. I think in Indonesia you might have to ask FDA.
I see. Okay. So one more question for Professor Konkanok. Can you brief the legal aspect of cannabis dosage form development? That is the question.
The legal aspect of cannabis
dosage form development.
or doses, right?
Dosage, yes, dosage.
Dosage form.
Dosage form.
Sorry,
Yes.
legal. Oh,
Yes.
okay. Legal. In Thailand, in my around the world right now, there's a sublingual cannabis extract in the name of Sativex. Is contains CBD and THC 1 to 1. That's sublingual. And then some company developed the buccal spray as well. And for Epidiolex, which is CBD, I think I'm not so sure. I think it's sublingual as well. So the thing is for dosage form, cannabinoid, both THC and CBD have very low bioavailability. So in oral dosage form, you get only 10% of cannabinoid. That's why people tend to use by sublingual route or oral by mucosal, and also vaporization or by smoke and what else, and yeah, so that the cannabis will go to the body, to the blood without the first pass metabolism. But if you talk about the law in Thailand right now, everything is possible. I mean, if you can register it. So in Thailand right now, we have some dosage form under the agreement of kind of Thai traditional medicines, because in Thai traditional medicines there's some recipe that contain a part of cannabis. So because the government try to encourage people to use it for health benefit, so they kind of try to encourage the traditional medicine uses. So there are the dosage form of tea, and then the capsule, and also ointment containing some of cannabis, but in very small dose, very small concentration. So we start to use this traditional medicine from I think the beginning of this year, and we always monitor the side effect, but there's very low side effect because of very low dose. So for example, when I did the analysis, there's only something like 0.3 of THC in the dosage that people take. It's very low concentration, because for Thai traditional medicine it contains many other herbs.
I see. Okay, that's great. One last question for this session. Professor Kolkanok, apart from cannabis female plant, is there any therapeutic effect on male cannabis plant?
Okay, that's very good question. Right. I don't know. Sorry. Let me put in this way. In our Thai traditional use, sometimes we use leaf. So we don't care if it's female or male. So we just collect leaf and make a tea, and then it helps to help people get sleep better, something like that. So I think there are therapeutic effect even, but the level of THC, I mean, all cannabinoids of male cannabis is lower than female flowers, because of you know that cannabinoid produced in the trichomes. So in female flowers, there's a lot of trichomes, so that's high THC and CBD. But in males, there's trichome as well. So you can use it, but the dose is kind of lower. But for Thai traditional use, we use very low dose anyway, so we don't care if it's male or female, right? But if you want to have a very kind of medical product, like in kind of Western way, then you need to standardize. And right now we are doing research on hemp, and we're gonna have the samples of female and male flowers of hemp next year, in the beginning of next year. So I might be able to answer you more of that by that time.
Okay, Professor Kolkanok, I definitely will visit you and we will have tea together. We will have tea together.
Yes, with gansha leaf.
Okay, ladies and gentlemen, this session is coming to an end, but I strongly believe that all information that we gather through this session will be very beneficial for developing further collaboration among us. Even though we are in a social distancing during the corona pandemic, but we are still connected through such virtual conferences and other activities which enable us to be always together. So I believe that this conference definitely can help us to work hand in hand for a better future, especially in pharmacy and health sciences. Thank you very much, speakers and audience, for your cooperation and participation. Please enjoy the whole session of the conference. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Professor Mangestuti, for moderating the first speaker session magnificently. Can we give big round of applause to the speakers and moderators for this first session? Congratulations, and thank you so much for the insightful discussion in this very morning. Now, we would like to proceed to give a token of appreciation to all of the speakers and moderators for the first plenary session. The certificate will be given by the Dean of Faculty of Pharmacy, Universitas Airlangga. To the first one, we would like to give a token of appreciation to Dr. Randa Apoteker, Raden Roro Maya Gustina Andarini, Master of Science, from the National Agency of Drug and Food Control of Indonesia. Thank you so much. And for the next one, we would like to give certificate of appreciation to Professor Dr. Habibah E. Wahab, Fellow of the Royal Society of Chemistry, School of Pharmaceutical Sciences, University, Sains, Malaysia. And last but not least, for the third speaker, to Associate Professor Kornkanot in Kaninan from Faculty of Pharmaceutical Sciences, Nariswan University, Thailand. And also, we would like to give a token of appreciation to Professor Dr. Apoteker Mangestuti, Magister Science, from Faculty of Pharmacy, Universitas Airlangga, as the moderator for the first session.
Thank you very much.
Thank you, Ibu.
Thank you to all the speakers and moderators. Ladies and gentlemen, now we have reached the end of the first session. Coming up next, we are going to have a parallel session for the poster presentation. For those who join the parallel session, we ask all the participants to log out from the Zoom account and then please log into your Zoom account according to your session. For e-poster presenter, please check the Zoom meeting ID and the passcode sent by the committee in your email or in the ICPHS website. Please be noted that the first poster presentation session will be started at 11:25 and the second session at 13:25 Western Indonesia time. The presenters should join the room at least 10 minutes before the presentation begins. Please rename your Zoom account according to the format. For poster presenter, room number, _ session number, _ poster code, _ name of presenter. And for the participant, the format as follows: room number, _ name, or as can you see on your screen. We will see you again at the second speaker session. Have a good time for your presentation. Thank you for attention and we will see you again this afternoon. Wassalamualaikum warahmatullahi wabarakatuh. Vaccination advocacy, educating and ensuring vaccine safety. Without any further, ladies and gentlemen, please welcome Cik Nur Hafsan Muhammad Hanafia.
Ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh and good afternoon. As we are about to begin, please be seated and make yourself comfortable. I have a few housekeeping notes to make before we begin the second plenary session. First, I would like to seek your cooperation in participating the whole session. Kindly mute your microphone throughout the plenary session to avoid any interruption. A quick reminder on the flow of the session. Each of the panelists will be given 30 minutes to share their topic of discussion, and we will have two Q&A sessions. The first Q&A session will be right after Dr. Wang's talk, and the second Q&A session will be after Prof. Bambang Prajogo. However, you may write down your question in the chat box, and we will read out during the Q&A session. Please write your question at the Q&A box by writing name _ ASK _ speaker's name _ question. So as an example, Hafi _ ASK Yiwei _ what is androgen? Thank you for your kind cooperation and attention. Dear respected plenary speakers, Dr. Yiwei Yang, Dr. Fatipatu Zahra Abdul Aziz, Prof. Bambang Prajogo Eko Wardoyo, ladies and gentlemen, it is my pleasure to be the moderator for the plenary session 2 this evening. I am Hafsan Hanafia, And on behalf of the third joint UNAR USM International Conference of Pharmacy and Health Sciences 2020 Committee, I would like to extend a very warm welcome to all of you. We appreciate you taking off your busy schedule and joining us today. We hope you will learn a lot today as we have lined up 3 distinguished speakers broadcasting all the way from Australia, Malaysia, and Indonesia that will be fruitful and engaging. So for the first plenary, I always like to use the phrase tak kenal maka tak cinta, or can be translated as to love somebody, you need to know somebody. Hence, let me tell briefly about our first plenary speaker, Dr. Yi-Wei Wang. Dr. Yi-Wei Wang obtained her PhD from Kingston University, United Kingdom in 2007. Currently, she is a Senior Research Fellow in Surgery at the Concord Clinical School, University of Sydney. She is also a Senior Research Scientist leading the Wound Healing and Skin Regeneration projects within the Burns Research Group, Anzac Research Institute, New South Wales. Her initial work focused on production of a novel polycreploxan scaffold for drug delivery. Dr. Wang's research into skin regeneration using synthetic biomaterials and advanced biotechnology has contributed significantly to the field of skin tissue engineering. Without further ado, I would like to invite our first plenary speaker, Dr. Yi-Wei Wang, and share with us her expert views on androgen and severe burn injury. Please welcome her.
Thank you. Thank you for the introduction. Now I just go to share my screen first. Okay, so I'm very honored to be here, and then thank you for the committee for the conference who invite me to give this presentation. As the introduction, I have the pharmacy background and I did my PhD in pharmaceutics, mainly about like drug delivery and tissue engineering. And after my PhD and working 2 years in Queensland Uni, I joined Anzac Research Institute with the interest of the wound healing and the severe burn injuries. So today I would like to give you a topic about androgen delivery and severe burn injuries. So what is burn injuries? Burn injury can be defined as the damage to skin tissue caused by heat, cold, electricity, chemicals, or radiations. And then burn injury can be classified into 4 different degrees, such as superficial, that like a sunburn, you don't need any treatment, it can help heal by itself. And then superficial, partial thickness, and the deep partial thickness, normally the wound turn to be this white and cherry-ish colors, or the full thickness burn injury, which is the injury can be done to the muscle or even to the bone. So normally for the burn injury have like superficial, deep partial thickness to full thickness, or the patients require the surgical treatment. So some WHO statistic, roughly around 6.5 million people got a burn injury each year, and around 180,000 people died. And the burn injury is the second most common injury in developing countries. So the challenges in a burn wound management. So the most important is the local wound. So we need to treat the local wounds. And then, however, severe burn injury also trigger the what we call hypermetabolic response. So when a patient have the burn injury, normally they trigger the glucocorticoid level, which is the stress hormone. So all the patients getting very stressed, and then they have double or even triple their energy expenditures and then lose their weight. So what we know from here, when the patient lost around 10% their total body weight, they normally impair the immunity. And then if they lost over 20%, normally the wound healing is delayed. And then if patients lost over 40% of their body weight, the death or the mortality rate can be 100%. So at this stage or at this time, for treating the burn injury can be 2 arms. It's like a local healing and how we attenuate the hypermetabolic response. So the current treatment is the golden standard, which is the split thickness skin graft. So what we do is, as you can see from this photo, we harvest a healthy skin taken from the donor size, and then we measure it to increase the size of the area, and then we put onto a clean wound. So for some patients have over 50% of the burn injury, they need to undertaken this surgery for multiple times. And then there are many other treatment for the severe burn injury, such as the non-pharmacological via nutritional intervention and the early exercise. Pharmacological, so far we know A has the clinical trial for human growth hormones or using the insulin therapy or with the anabolic androgens called oxyandrogens. So what is androgens? So androgens promote the development and the maintenance of the male characteristic. And the testosterone and the dihydrotestosterone are the main androgens in both male and females. And then in two of these, testosterone is the predominant circulating androgens. And then testosterone can bind to the androgen receptor and also serves as the obligate precursor for the estradiol, while the testosterone, which is DHT, only bind to the androgen receptor. So what we know about the role of androgens in wound healing? So there are many previous studies have studied the androgens. So what they found, both testosterone and DHT shown to have inhibitory effect on the local healing process in both mouse study and the human studies. And then the scientists find like the testosterone can increase the local inflammation and then reduce the reepithelialization, also reduce the keratinocyte proliferation, and that with a slower wound healing. However, another interesting finding in the previous study is when clinicians or surgeons use the oxyandrolone, which is the synthetic anabolic androgen, they found totally opposite effect for the burn injury patients. So the clinical trial showing that oxyandrolone can better maintain the lean body mass, and then they can improve the protein synthesis, they can shorten the length of hospital stay, and improve the long-term recovery. Although there is a disadvantage of hypertoxicity, but oxyandrolone has been widely used in the US in the burns unit. So that raises research questions. So what is the exact role of androgens in both cutaneous wound healing and the burn injury? Why they have totally opposite reactions? So this lead to our research. So the aims of our research study is to identify the role of androgens in specific stage of the wound healing post-severe burn injuries, such as inflammation, cell recruitment, cell proliferation, and the matrix remodeling. And the second aim of this study is we want to local delivery androgens to examine if local delivery of androgen also have the similar positive effect on the local wound healing. So this study also to develop and characterize a novel wound dressing by using polycaprolactone and incorporating with the pure androgen DHT or anti-androgen flutamide for local drug delivery. And then we also want to investigate whether the local modification of androgen actions has any impact on the burn injury wound healing. So, for our experimental design, we used the biopsy mice, roughly around 12 to 16 weeks old, and each of the mice received this 2 times 2 centimeters square burn injury, roughly equal to 30% of their total body surface area. In our recent published paper, we consider 30% of TBSA can be the severe burn for animals. And then in 48 hours, all this damaged skin will be debrided, which is just being removed, and then will be dressing up. So housing, handling, pain relief is all approved by the ethics committee. So in the experiments, all the mice has been randomized to 2 groups, which is the control group or the DHT treatment group. So in the DHT treatment group, mice will receive this silicone tubing capsules showing like this, which contain the DHT powder in it. And then the DHT, the silicone tubes can produce a very stable blood DHT level around 1 to 1.590 gram per ml until 1 year, which is enough to cover like the whole healing process in animals. And after the burn injury and the treatment, we have been examined like the wound healing rate using the VisiTrack digital system. And then we also measured 4 overlapping local healing process, which including inflammations, reepithelialization, cell proliferation, and the remodeling. So our results shows that when the animals receive the DHT treatment, there is a significant increase in wound healing, particularly on day 5 and then day 7, as shown in these photos. And histology images also confirm the finding. So for people not really familiar with histology, you can see this is the normal skin structures. You can see the epidermis, dermis, and then some skin appendages. And then on this area, there's all the wound. So you will see the inflammation. This is the muscle underneath. So in comparison, we can see in the control group, there is a very huge or severe wound, which we can even see the other edge of the wound. And then after the animal have the DHT treatment, the wound is much, much smaller and then can be seen from here. So after that, we measure the inflammation by using the real-time PCR. And then for both L6 and the TNF-alpha, what we found is on day 7, which is in the early stage of the healing process, both DHT group have significant increase of the inflammatory response locally, but there's no differences on TGF-beta1. So after that, we measured the cell proliferating using an antibody called PCNA, which stain all the positive cell which is proliferating. So the quantification by 30 cells, we found like both the DHT group on both day 7 and day 14 has significant faster cell proliferating. And in the remodeling phase, which is the collagen deposition, when the wound is like 90% to 100% healed, this collagen will be regenerated to refill the gap. So what we can see again, the DHT group has a much higher collagen deposit. And then you can see from these 2 images that in the DHT group, the color of the green, which is showing the collagen fibers, is very similar to the normal skin. However, in the control group, you will see this all purplish, which is still showing very high inflammatory response. So after the local assessment, we have also conducted the systemic assessment. The reason is we know like androgens being known as anabolic hormones. So all these positive results is expected to be seen through the systemic effect. So that's why we measure the body weight. We also measure the body composition by the DEXA scan, and then all the tissues has been harvested and collected, which including blood, skin, wound, liver, kidney, spleen, all the fat, the brown fat, white fat, different muscles, and the seminal vesicles, testes, and kidney. We also used a very new state-of-the-art called a metabolic cage to measure the metabolism change of the animals. So this cage gives us 24/7 data. They measure the oxygen intake every 5 minutes. They measure the animal's movement by night and daytime. And then we also measure like the cytokine and chemokine using the bioplex. And then the immune cells population was detected by using the flow cytometry, which we want to see the big picture, how the androgens affect on severe burn wound healing. So our data showing that when the animals receive the DHT treatment, they have better maintained their body weight. As you can see from 100% right after the burn injury, both of group of animals drop their weight, but DHT better maintain their body weight. And then by using the DEXA scan, which we can measure both the total lean mass and the total fat mass. We can see in both groups, there's no lean mass loss. They all remain around 5% to 10% positive. And however, both group has lost roughly around 5% to 12% of their fat mass. The reason is when the mice has the open wound, they normally lose their body heat or body temperature. So they are burning their fat to keep themselves warm. That's the reason we see this significant drop of the fat mass for both groups. However, DHT doesn't have any effect on reserving the lean mass or the fat mass. And then by analysis all the data from the metabolic age, we found like after the burn injuries, both group of the animals had significant increase of the energy expenditures and then reduce of the movement. Then mainly because they have the pain or uncomfortable from this severe large wound. However, DHT seems has no effect on either energy expenditure at daytime or nighttime or any effect on the movement. So we get this negative results because we expect androgen as an anabolic hormone should have a positive effect on this metabolism, but all the results saying no, DHT doesn't have any impact on the metabolism. So the reason is why androgens have promote severe burn wound healing. There must be a reason. So we try to dig out more about how androgen react or how androgen modify the burn injury wound healing. So during the harvesting of all the organs, one of the most interesting observation is the size of the spleen. Sorry. So as you can see from these images, this is a normal spleen from the mouse. And then after the severe burn injuries on day 7 and then day 14, you will see like the spleen enlarge their size roughly around 2 to 3 times. And then we measure the weight of the spleen percentage to the body weight. We can see like in the DHT treatment group, the increase of the percentage on day 7 and after that they dropped. However, in the control group, the peak was observed on day 14 and then gradually reduced. So that's suggesting or indicating that DHT may regulate the inflammatory response systemically because the spleen is known as a very important organ for systemic inflammations. So after that, we did the histology analysis on the spleen. So what we can see is after burn injuries, there is the disorganized splenic structure of the spleen, and all these red pulps, the white pulps infiltrated into the red pulps, and then you can see there's a multinuclear granulocytes stored in the spleen. However, in the DHT group, DHT preserved normal splenic structures, and then you can see this is the white pulps and then the normal red pulps. So that's a further confirm that like DHT may regulate or may promote the burn injury wound healing through the systemic inflammation. So that's why after the histology analysis, we conduct this multiplex. So multiplex is like a multi-ELISA, which screen all different cytokines and chemokines in one kit. So in our kit, we have 22 inflammation-related cytokines has been detected, which including all the pro-inflammation, anti-inflammation, and all the chemokines. So our data suggests, or our data shows that both L6, L12 has significant increase on day 3 only in the DHT group, as you can see here, and also L1 alpha, 1 alpha on here. And then like for the MCP1, which is secreted by the monocytes, we also see this significant increase of the concentration observed in the DHT treatment group. So this, all the data we have seen so far, the change of the spleen, the histology observation of the spleen, and also the multiplex data suggests, yeah, there must be something changed in systemic inflammation. So after that, we measured the immune cells population in the blood. Like what we hypothesized, both of the lymphoid cells and the granular size has increased on day 7, even though this is not significant differences, but you definitely can see like a trend of increase on day 7 in the DHT group, right? And after that, we will see like a significant drop of these immune cells on day 14. And after that, we measure all the splenic immune populations, which include the lymphocytes, B cells, T cells, and also the myeloid cells, which is the lysis-6 monocytes and the splenic macrophage. One of the most interesting findings are here. We do see an increase of the monocytes on day 7 and significant drop on day 14. At the same time, we see a significant increase of the macrophages. So that indicates that the bone marrow produce the monocytes in the circulation and then store them in spleen as a macrophage. As we know, like a macrophage only be differentiated from monocytes, and then they cannot be produced in the local wound area. So we want to see if this circulation change of monocytes and the macrophage has any contribution locally to the wound healing. So after this study, we stained F480, which is the antigen for macrophage. So we stain roughly this area, which is the edge of the wound, and then this is the normal skin as the control. So you can see these images showing the normal skin, and then you will see very low number of the macrophage, which we call they are the residue macrophage, just sitting there. And then in the control group, you can see like after the burn injuries, there are very high or intensive staining of the macrophage from day 3 all the way to day 21. However, in the DHT treatment group, it seems that there are more macrophage has been detected or seen on day 5, day 7, and then after that, they gradually reduce down. So that's indicating that there are more macrophage in the circulation going to the wound area to clean the debris or clean the bacteria from the wound, and then which accelerate the whole turnover of the healing process. We have also done, quantification of this F480. Like we expect, we do see an increase of the F480 quantification on day 5 and day 7, and a significant drop on day 14 and day 21. So in summary, like the DHT treatment has significantly promoted the burn injury wound healing and better maintain the body weight. And then they have no effect on metabolism, which is disappointing or not disappointing, but what we found, they better preserve the spleen histology. And then DHT treatment shortened the inflammation phase over the whole healing process. So this is the mechanism we try to figure out behind, even though we haven't touched every single area, but we think this is the big picture. So what happened is, when we give the DHT systemically, they bind to the antigen receptor on the macrophage, neutrophil, and monocytes. And they secrete all these cytokines that we see from the multiplex, and then they get into the blood vessel, get into circulation, and start into the spleen. And now all this increased macrophage finally recruited to the local wound area and then faster clean the wound and then accelerate the turnover from inflammation to proliferation to remodeling. So the role of DHT in severe burn injuries is they change turnover. However, and this study just being accepted and published in the International Journal of Molecular Science. So if anyone interested in our study, you can go and find these papers, and there are more information in it. So when I talk to the surgeon or clinician by using a systemic DHT treatment, they always have a concern because hormones or steroids, they always have the side effect. So that's why in the AIM2, we were thinking if we can create a novel like a wound dressing that can control release either androgens or anti-androgens in treating severe burn injuries. So the AIM2 is about the local delivery. So we expect that blank, which is the normal PCL electron spun scaffold, have the normal healing. When the scaffold contains the DHT, they may delay the healing, we don't know, or local negative effect or systemic absorption, excuse me, or like a flutamide may faster the wound healing or reduce the inflammation, like people have been reported in the cutaneous wound healing. So all the scaffold is made from polycaprolactone, which is FDA-approved biomaterials, and then it also has been widely used as a controlled drug delivery device. And the drug loaded the scaffold were produced using the electron spinning, and then this is how the scaffold look like right after they've been peeled off from the target here, and then there's two SEM images showing like all the drug has been well encapsulated in the polymers. So in vitro drug delivery or release was detected in a PBS at 37 degrees, and the concentration of DHT or flutamide was measured using the LCMS, which is the mass spec. So we can see like DHT release very constantly, roughly around 30 to 40 microgram per mil from day 0 to day 21. In comparison, flutamide released roughly around 15 microgram per mil all the way to around 12 microgram per mil until day 21. And these two SEM images is after release. We can see like the scaffold remaining their porosity and structure, even though it seems a little bit flattened, but it doesn't have any significant effect on the structure. So after we create this wound dressing using a polycaprolactone, another question is, when is the best time we apply this dressing? So we know like when you have an open wound, if you apply any drug on it, it will be very quickly absorbed systemically. So that's why we want to see if the drug gets into the circulation or not. By validating this, we use the DHT loaded PCL scaffold, and then we design two different experiments. So we either apply them on day 0, right after the debridement with the big open wound, or apply them on day 7. Day 7, when there is a very thin layer of epidermis. And then in this validation study, we use the castration mice, which the testis has been fully removed. And all these mice has a very, very low endogenous androgen levels. So the results show like, when you apply this scaffold on day 0, there's a huge increase or significant increase of the DHT levels on both day 3 and day 7. And however, if you apply this on day 7, thereafter, there's a very minimal detectable DHT in the circulation. The maximum we can measure is roughly around 0.5 microgram per mil. So that's why we determine like the scaffold will be applied on the wound area from day 7. So when we put the wound on day 7, we do see like flutamide accelerated healing, particularly on day 14 and day 21, roughly around 20 to 30% increase. And then the histology images further confirm the length of the wound. Like we can see the DHT loaded scaffold have a much bigger wound compared to the flutamide treated scaffold. And then we also measured the length of this called reepithelialization, which is the major mechanism in human wound healing. But in mice, they also the combination of contraction and the reepithelialization. So like we expected, the flutamide group has a much longer length of the reepithelialization, which confirm like the better healing is from this migration of the keratinocytes on the surface of the epidermis. And then thereafter, we conduct this PCA again, But surprisingly or interestingly, we see like the DHT scaffold actually have the higher PCA positive cells. This suggesting that DHT can accelerate the cell proliferating, but may not have any effect on the cell migration. And the collagen deposition was a further analysis using the Masson checkpoint staining. All this green color showing the collagen bundles. And then you can see in the in the flutamide scaffold group, they are much more or significant more collagen deposition compared to the DHT group. And after that, we want to see even local treatment has any negative or positive effect systemically. So even though the DHT only locally released to the wound area, but they still better maintain their body weight, especially on day 14. And then this increased the body weight may due to increase of the food intake. Like when animals heal faster, they will be like their stress hormone will be reduced and then they will start eating more. And then that may be the reason why there's a gain of the body weight. And all this local delivery from scaffold seems has no effect on the androgen-dependent organs, which is the kidney and the testes. You can see all the three groups has a very similar weight of the kidney and the testes, both on day 14 and day 28. So in summary, what we found in local delivery is the flutamide PCR electron sponge scaffold significantly accelerate the brain injury wound healing. But DHT PCR scaffold has similar wound healing compared to the black scaffold. And then also the DHT PCR scaffold promote the cell proliferating of keratinocytes and then better maintain or remain the body weight. And then no side effect have been observed over 28 days in all different groups. So in conclusions, the systemic treatment of DHT facilitates the local healing by accelerating the resolution of systemic inflammation, but not through the alteration of post-burn hypermental body response. And then the local delivery of flutamide via the electron sponge PCR scaffold promote the wound healing after severe burn injuries. So all these exciting findings open a very novel therapeutic avenues in treating severe burn injury with the combination of positive effect from the systemic DHT treatment to the local flutamide treatment. So finally, I would like to thank my team, my mentor, Professor Peter Mays, who is the leader of our research group, and my PhD student, Dr. Peter Shi, who just been awarded the PhD, who conduct all the experimental in vitro and in vivo, and my research assistant, Roxanne Francia, and the Australian National Health and Medical Research Council for the foundings. And then thank you, everyone. Thank you.
Thank you so much for the great sharing, Dr. Wang. It is surely great finding to see how androgens, a sex steroid hormone, plays an important role in the response of body system to trauma, especially in burn, and how androgen can modify the specific wound healing phases such as the inflammation, cell recruitment, and matrix remodeling. So now, I would like to start the discussion session by reading the first question asked by our participant. So the first question comes from Amina Yusuf. So she is asking, there are a number of medicinal plants with wound healing effect. Can we equally replicate such studies using such plants? So this is called for a collaboration, ma'am. So I think you might need to contact her later on. So what would be your answer then?
Yes, definitely, because this model has been well established and validated, and then we have published these models with all the details of how we create it. I think the best way to include or incorporate the plants is like if you can extract the active component from the plants and then load it with either like different formulation. It can be cream or it can be like scaffold, and then test on this severe burn injury wound healing models. Yeah, welcome to contact me. I will leave my email address in the community. Yes.
That would be great. Okay, so we will go for the next question, which is from Mr. Suharjono from Faculty of Pharmacy, UNAI. Do you think safety androgen topical for severe burn injury or regeneration new skin? I think it's more like,
Well,
do you think it's safe or not, right?
well, this is a very interesting questions. So it depends on who you ask. If you ask endocrinologists, they will tell you yes. And then if you ask the burn surgeons, they will tell you no. So we still try to understand if it's safe to use and what is the safe dosage to be used. So oxyandrolone is the only androgen has been used in treating burns, but this is the oral formulations. So we haven't seen any topical formulation has been used in a burn injury. So we don't know.
Okay. I have a question. Are you familiar with the tilapia skin grafting?
Yes.
Yeah, okay. Have you ever in a future plan like trying to compare whether is there any difference or like is there any benefit of using an androgen rather than using that tilapia method?
But the tilapia method is not allowed to be used in Australia at this
Okay.
stage. We have a collaborator in Sydney Uni, but we have been trying to use the skin for the koala because every year in Australia, you know, there is a bushfire. So there are many koala has been severely injured. So we try to import the tilapia fish skin and see if it worked.
So one question to raise is that what would be the main concern of complications used for androgen, especially because it's more of like a gender related, right? Especially if, example, in your study you would use more of male mice and men in particular. But how about for female? Do you put into such concerns of any complications?
Yes, we do. I think major is the hepatotoxicity and also the gender change. But however, during the severe birth, the whole circulation will be changed. As you can see, like they're in the hypermetabolic state. So even though you give them like different steroids, it doesn't mean they have a later side effect. But the answer is no. So one of my other PhD students still working on the female, and I think we will answer that question much better, like how they change the period, how they change the weight, the breeding time, everything.
Is there any questions from the participant that you would like to highlight? Because right now I'm only like receiving only 2 questions at the moment. But actually I would like really want to know what is your future directions in ANZAC Institute, like what is your future directions of the androgen usage in burn or wound healing, which means like how would you see that the usage of it in the future?
We encourage, or we've been encouraged to try the clinical trials. So one of our, the director of our ANZAC Research Institute is endocrinologist, and he is the expert of androgens. So he definitely confident about using the androgens in a burn patients. And then the future direction for our group, we have like different projects. So this is one of the major project has been funded by NHMRC, and it's almost done for male. And then we currently working on the female, especially the side effect part. We also have a project to study the glucocorticoids, which is the stress hormone regulation in wound healing and scar formation. And also we have few different projects in tissue engineering, which using all the thermosensitive gel or using different scaffold to treat the burn injuries.
Okay, so yeah, it's really quite interesting to actually see that for me, this is something new about local wound healing by using sex hormone, where you see that in your own conclusion, you say that DHT treatment will facilitate local wound healing by accelerating the resolution of inflammation, but not through alterations of post-burn hypermetabolic response. Maybe in a way that when you see a burn patient in the hospital, right, they will more focus on the management as well, like giving all of the nutritions via TPN and so on. So yeah, thank you very much, Dr. Wang. That is a very good sharing that you have done there. I really appreciate it, and I do hope the participant appreciated it. So before we bid farewell to Dr. Yue Wang, on behalf of the ICPHS 2020, we would like to present the Certificate of Appreciation to Dr. Yiwei Wang, who has presented by Prof. Habibah A. Wahab, which is the Dean of School of Pharmaceutical Sciences, University Sains, Malaysia.
Thank you.
So thank you so much. Please give her a group of applause. I hope that you can see that I'm clapping from here.
Thank you,
And we do
everyone.
hope there will be future collaboration, okay? Thank you so
Yes,
much.
definitely.
Have a great day.
Same to you.
So now, thank you to all. But first, it is not the finishing. We will still need to go for the next plenary speaker. So let me briefly introduce you to Dr. Fatimah Zahra binti Abdul Aziz. So she obtained her PhD from University Sains Malaysia and is currently serving as one of the clinical pharmacy lecturers in School of Pharmaceutical Sciences, University Sains Malaysia, which is the first pharmacy school in Malaysia. Her involvement in teaching focuses a lot on Malay traditional medicine, which is incorporated in the B.Pharm curriculum. Her research interests and expertise are pharmacometabolomics, nephrology, clinical pharmacy, nuclear magnetic resonance, traditional and complementary medicine. Aside from teaching, Dr. Fatimah Zahra is also one of the fellows for the USM Entrepreneurial Incubator and involved in the commercialization of the Musafir and Travel Kit, which is produced by the Family Wellness and Beauty Kiosk. She is also a keen public speaker, educating the public on the safe use of traditional medicine, supplement, and cosmeceuticals. Her passion towards Malay traditional medicine had allowed her to be one of the certified Malay traditional practitioners, specializing in Malay massage. We will now let her share her views and insight on the traditional and complementary medicine practice and education in Malaysia. Please welcome her.
Thank you, Ms. Moderator. So allow me to share my screen first. Okay, can everyone see my screen? Okay, can you hear me? Okay. Okay, Assalamualaikum and hi everyone. I'm Fatimah Zahra Abdul Aziz from School of Pharmaceutical Sciences, University Sains Malaysia. Okay, first of all, I would like to express my gratitude for the opportunity given in this conference to share about traditional and complementary medicine in Malaysia. So for the next 30 minutes, okay, I guess, I will be sharing with you about the progress of traditional and complementary medicine, which I will address as TNCM after this, focusing on practice and education in Malaysia. Okay. Okay, my sharing session will be divided into 4 parts. Okay, I will start with a brief introduction on TNCM, including some definition. Then we move to TNCM practice in Malaysia. And the third part, I will share about education. And lastly, about the future plan for TNCM health services in Malaysia before I end up my sharing session. Okay, TNCM is now one of health service that acknowledged under the WHO, okay, World Health Organization. It been recognized into the mainstream of healthcare. Okay, people around the world start to accept the effectiveness of the TNCM services, as we can see from the report. Okay. Traditional and complementary medicine, TNCM, is an important health service, but often underestimated. Okay, this is the reason in 2014, WHO came up with WHO Traditional Medicine Strategy for 2014 until 2023, which they focus on policy, safety, efficacy, and quality. Okay, they also look into the accessibility of the services, by increasing the availability and affordability of the services, especially for bottom billion population. Okay, let us look into current practice of TNCM worldwide. Okay, this data, I get this data based on WHO Global Report on TNCM 2019. We stated out of 170 member states, 88% of member states acknowledge their use of TNCM. Okay. Sorry. Okay. This means the country involved formally developed their policy, laws, regulation, program, and office for the TNCM division. And Malaysia are one of the member states. Okay, now let's move to the second part. I'm going to share on development and current TNCM practice in Malaysia. Okay, this is Malaysia. Okay, a quick definition on traditional medicine and complementary medicine. Okay, traditional medicine is a knowledge, skill, and practice based on theories, belief, and experience of local native people of that particular country. And they use it to maintain the health as well as in prevention, diagnosis, improvement of treatment of physical and mental illness. Okay, meanwhile, for complementary or alternative medicine, is a healthcare practice that is not part of the country's own tradition or conventional medicine, or we also known as modern medicine, current medicine. Okay, I give you one example. Let's say we took massage therapy as an example. We know that there is a lot of massage therapy around the world. To name few, we have shiatsu massage, Swedish massage, aromatherapy massage, and reflexology massage. Okay, and here in Malaysia, we have Malay traditional massage. Malay traditional massage is categorized under traditional medicine in Malaysia, and it is a skill or practice of native people in Malaysia. Let's say if we integrate shiatsu massage in Malaysia healthcare system, it will be recognized under complementary medicine definition in Malaysia, as we know that shiatsu massage is origin from Japan. And it will be vice versa in the Japan, which let's say shiatsu massage will fall under traditional medicine definition in Japan, and if we implement Malay traditional massage in Japan healthcare system, it will fall under complementary medicine definition. Okay, this example explains why the health practice included under the term of TNCM can vary from country to country and from region to region. Okay. In Malaysia, traditional and complementary medicine is a form of health practice designed to prevent, to treat, or to manage illness or preserve the mental and physical well-being of an individual, exclude medical or dental practice used by medical or dental practitioners respectively. This is the definition that being defined in our TNCM Act 2016. Okay, I guess we have some times. Can we do some activity? Moderator, shall I? So because we can't do any exercise, so I hope that this simple activity will refresh our conference. Okay. Okay, what is the first thing that come across your mind about type of TNCM? Can you please go to www.mentimeter.com and use the code 4175635 and fill in what is the first thing that come across your mind about type of TNCM. Can you see the activity? I also list the practice that available for TNCM for you to choose. Okay, I can see one.
Yes, I can.
Anyone else? Acupuncture, jamu herbs. 5, okay. Some more?
Maybe all the participants can actually put the list that you actually even tried, like I did.
Yeah, all the participants. Can they see? The participant can see this, right?
Yes, I can see.
Yes, I can see.
Okay. Please go to mentimeter.com and please type 4175635.
Yeah, already.
Only 19. How many of us?
9, I think.
For this session, including the participant? 19, 20, still waiting. So jamu is leading. Jamu herbs.
9.
Ayurveda, I also one. Homeopathy heritage, okay. Waiting some more. Maybe another 2 minutes for this activity.
Dr. Fatima, I've already put the link, so anyone can just click the link, so that will be directly to Mentimeter.
Okay, thank you, moderator, Miss Moderator. You can click the link provided by the moderator. Some more. Another 1 minute. Okay, there's still
Jamu. Jamu, yeah.
Jamu. Jamu is leading. Jamu
Yes.
and okay. Okay, thank you all. Okay, we go back to our okay. You still can fill in the Mentimeter and we will see the result after this, okay? Okay. So after the definition, okay, we already done with this, okay. The Minister of Health in Malaysia has taken a positive and proactive approach towards the NCM to ensure its quality and safety for the consumer. So that's why we came out with the national policy of the NCM, which stated that TNCM shall be an important component of the healthcare system with will coexist with modern medicine and contribute towards enhancing the health and quality of life of all Malaysians. And they also have a vision of that TNCM shall optimally be integrated in the Malaysian healthcare system to achieve a holistic approach towards enhancing health and the quality of life. Okay, I will go into a brief history on development of the NCM practice in Malaysia. Okay, let me take you back into time. It started off with a research proposal for alternative medicine back in 1987. Okay, and in 1996, a TNCM unit was formed under the Family Health Development Division, and 2 years later, TNCM standing committee was established with a big responsibility to develop and incorporate of the NCM into a national policy. Okay, entering year 2000, Health Medical Research Center was formed under Institute of Medical Research, also known as IMR. Then a year after that, the first national policy on TNCM was launched, followed by the establishment of TNCM division in 2004, which we see it took 8 years after being formed under Family Health Development Division. Okay, then if move again in 2007, TNCM services start to be offered in public hospital, and first hospital offering the service was Kepala Batas Hospital in Pulau Pinang. In 2016, the Malaysia government come out with the Traditional and Complementary Act 2016, also known as 775 Act, which enlisted several recognized practice area under the same act. I will discuss about this later. Okay, coming back to 2020, we have 15 public hospitals offering the TNCM services. As the TNCM practice progress in Malaysia, the TNCM division with technical support from WHO come out with the TNCM blueprint 2018 until 2027. Okay, the planning. The blueprint aimed to facilitate the regulation, integration, and economic development of the TNCM industry in Malaysia. Also in this blueprint, the TNCM division has identified the challenges in TNCM industries, the general directions, and 4 focus areas which are practice, education, medicinal materials and product, and last but not least is research area. And from the same blueprint, the committee came out with a strategy and action plan to be implemented in the next 10 years for TNCM in Malaysia. Okay, in order to maintain the quality, safety, and development of the TNCM service in Malaysia, Ministry of Health also have regulatory divisions that will monitor and guide any related TNCM matters. Okay, as an example, the product development of a herbal plants. Okay, the product anyone who want to produce the herbal plant will need to comply with these 3 acts, which is Sale of Drug Act 1952, Poison Act 1952, and Dangerous Drug Act 1952. Another example is for practice and practitioner domain. For this one is fall under TNCM Act 2016, and is governed by the TNCM Council and TNCM Division as the secretariat to the council. Okay, anything related to the practitioner and also the practice itself will fall under Traditional and Complementary Act 2016. And to ensure the quality of research, Malaysia have a National Committee on Research and Development of Medicinal Herbs under the National Institute of Health. Okay, as I mentioned earlier, a year after 775 Act implemented, the Malaysia government gazetted and published 7 recognized practice area in Malaysia. Okay, so we have traditional Malay medicine, traditional Chinese and Indian medicine, homeopathy, chiropractic, osteopathy, and Islamic medical practice. Okay. Each recognized practice area, this 7 practice area will be monitored by a designated practitioner bodies. Okay, for example, traditional Malay medicine will be monitored under Gabungan Pertubuhan Pengamal Perubatan Tradisional Melayu Malaysia. As for chiropractic and osteopathy practice, this area will be observed by Federation of Complementary and Natural Medical Association Malaysia. Currently, TNCM division under MOH has produced practice guidelines for TNCM practitioner to practice either in hospital setting or elsewhere in Malaysia. This guideline will help the practitioner to deliver a safe, standardized care for to the consumer. Okay, I'm showing in the slide here the available practice guideline for acupuncture, cupping, chiropractic, Malay massage, external basti therapy, and herbal therapy as adjunct treatment for cancer. Another practice guideline also available guideline on postnatal care, Islamic medical practice, and Shirohara practice. In 2018, WHO came up with a report on TNCM in primary health care. Other than China, India, and Korea, Malaysia is one of the countries that successfully integrate the TNCM service in public health care across the country. I'm happy to share. This is till date, 15 public hospitals in Malaysia is offering TNCM services. Public hospital, meaning under our government. Okay, for example, here in Kepala Batas Hospital, they are offering traditional massage, acupuncture, and herbal therapy as an adjunct treatment for cancer. We can see the positive direction of MOH, Ministry of Health, where more centers will be offering TNCM services in the future. Okay, this is type of services provided at TNCM unit in public facilities in our hospital here. We have Malay massage for chronic pain and post-stroke. It's indicated for chronic pain and post-stroke. Acupuncture service for chronic pain, post-stroke, and chemotherapy-induced nausea and vomiting. We also have postnatal treatment, also known as penjagaan pantang postnatal. It's well known as service for women after birth delivery. Herbal therapy is used as an adjunct treatment for cancer patients experiencing symptoms and complications of cancer and side effect of cancer treatment. And we also have 3 branches of traditional Indian medicine, include Shirodhara therapy, which is focusing on mental wellness, and followed by external basti and Barman therapy that focus on musculoskeletal problem. Okay, I would like to share our own experience in USM. USM has already offered the TNCM service as part of the healthcare for the patient. Okay, the service center is located at one of USM branch known as Advanced Medical and Dental Institute, or AMDI, which is in Bertam, Pulau Pinang. Okay, the TNCM service were operated under the MD Integrated Clinic. Okay. This is the building where we run the clinic that I'm showing you. from Monday to Friday, 8 to 5 p.m. Okay, as you can see, it's surrounded by herbs plant. A little bit about the clinic. Okay, the TNCM services started in February 2014, 7 years after Kepala Batas Hospital offered the TNCM service in their hospital. Okay, this clinic is headed by Dr. Ishaiful, who is a medical doctor with a master and PhD specializing in herbal medicine. All the certified practitioners who is practicing under this clinic have a medical background and working as registered medical assistant, nurses, and pharmacy assistant. Currently, we are providing services such as Malay massage and senaman tua. This is an ancient Malay exercise. Okay, these two therapy will be prescribed to the patient meant for treatment. Other than that, we also conduct a counseling services for effective and safe use of herbal medicine. The clinic also actively involved in teaching pharmacy students for TNCM courses. Next, we plan to expand the service by adding on a wet cupping and acupuncture. We're also moving towards to become a training center for TNCM in Malaysia. Okay, I would like to share with you a photo of staff that are responsible to run the clinic. Okay, if you look at this left side, okay, the guy at the front of at the front is the head of the clinic, Dr. Ishaiful, and with his team behind. And on the right side is the treatment room for the patient. This is a treatment room. And here's some more photo for okay, and you can see this is our staff in action. We are in the state of preparing a room for wet cupping services, as shown in the right side photo. Okay, what is the workflow of the clinic, the integrative clinic? Okay, the clinic already receives referral case coming from any registered health institution. The specialist will review the case before starting the treatment to ensure that the patient are suitable to undergo the treatment. Okay, patient will be prescribed with massage therapy, individualized to the patient condition. Then the patient will be assigned to the therapist based on gender. Male patient will be assigned to the male therapist, and female patient will be assigned to female therapist. Okay, each treatment will take around 1 hour per session, and it might require several sessions, repetition throughout the treatment course. So now we move to education in Malaysia. Okay, the TNCM education in Malaysia. Okay, in sustaining and maintaining the quality of the TNCM services, education and training are a crucial area needed to be strengthened. Okay, as mentioned in the Malaysia TNCM blueprint, this is one of the agenda. Malaysian government comes up with the standardized TNCM education system. The TNCM education system are divided into 2 certified programs, as you can see on the slide. Okay, for higher education program, the TNCM curriculum was developed in collaboration with Malaysian Qualification Agency, MQA, which is a governing body responsible for accreditation of higher education program in Malaysia. However, for the development and accreditation of TNCM skill training and certification, it should follow the standard by National Occupational Skills Standard. Okay, I'm sharing with you the list of university and institutions that offer TNCM higher education program. In Malaysia, the TNCM graduate and postgraduate program are being offered by local private higher education institution. Okay, this program has been accredited by Malaysia Qualification Agency, MQA. As for traditional Chinese medicine, we have PhD, Master of Science, Bachelor and Diploma program. And for the rest, such as chiropractic and homeopathy, we have bachelor program only. How about pharmacy program in Malaysia? As pharmacists, we play an important role in TNCM development. A basic knowledge in TNCM is a must for undergraduate degree program in pharmacy. Okay, in regard to that matters, okay, the Pharmacy Board Malaysia came out with a standard on approval and recognition of pharmacy program in 2018. We stated in the knowledge domain, okay, a pharmacy degree must ensure to have a course that teach the student about mineral and plants, and also traditional and complementary medicine. And pharmacy degree in USM leads the program by having 9 credits dedicated to pharmacognosy or ethnobotany and TNCM. This is out of the 145 graduation unit that we have. Okay, our program start to teach pharmacy students in second year with 3 unit core course. In this course, the student will learn about basic pharmacognosy and phytochemistry. Then in third year, the student will be taught on applied pharmacognosy as a 2 unit core course. And we also have a 2 unit elective course for Malay traditional medicine. And for final year, we have another 2 unit for traditional and complementary medicine, which will teach the student about philosophy and principle of traditional and complementary medicine practice that available in Malaysia. Okay. For final year student also, we have a field work where student is sent to integrative clinic in Namdi for 1 day attachment in a small group, basically 10 to 12 students per group, for them to explain on how the TNCM service will carry out. During the attachment, the staff will demonstrate on senaman tua, as you can see on this slide, Malay massage, acupuncture, and wet or dry cupping as shown in the picture. This one is for wet cupping and also dry cupping. Later on, the student will have a case study on actual TNCM cases from the clinic to be discussed later in the class. Okay. So I'm moving to the final one, TNCM direction and the future plan. What is the TNCM future plan in Malaysia? Of course, this is what TNCM division are working on, moving TNCM towards evidence practice. And next, we are going to have, hope to have the TNCM practice guideline as an evidence-based practice from consensus-based guideline. We also are going to enhance the research collaboration with the MOH and international and, Okay, till now Malaysia already have MOU with 5 countries, with China, India, Thailand, Brunei, and Indonesia. Okay, and more to come inshallah in future. With that, thank you for your attention. So I return the session to Ms. Moderator. Maybe we can also share again the Mentimeter to see the result. What is the final result? Okay, 44 of you share. Still, Jammu is the leading. Okay, thank you, Ms. Moderator. I just return the session to you.
Thank you so much, Dr. Fatimah Zahra, for your insight on education and practice of TCM in Malaysia and also the interactive usage of Mentimeter. So such a great setup clinic you have there, Dr. Fatimah Zahra. Personally, I myself am into and utilizing the chiropractic service and aromatherapy. And I think for myself is that teaching purposes, in terms of teaching purposes, I would like to know more on the effective usage of TNCM in cancer patient. Because as what mentioned by Dr. Fatimah Zahra is that in concordance with the World Health Organization policy and National Health Policy Malaysia, it has its own traditional and complementary medicine blueprint that includes the 4 components of practice, education, training, raw materials, products, and research. So before we allow for the Q&A session, we will first continue to our final speaker for his well-known effort in researching and publicizing the use of herbal medicine, Prof. Dr. Bambang Prajogo Eko Wardoyo. So let me briefly tell you guys about him. Okay. Prof. Dr. Bambang Prajogo Eko Wardoyo obtained his PhD in 2002, entitled Pengaruh Anti-Fertilitas Flavonoid dalam Justisia Gandarusa terhadap Epidermis Spermatozoa from Universitas Airlangga, and later serves as the professor at the Department of Pharmacognosy and Phytochemistry, and also served as the head of master programs of Science in Pharmacy at the same Faculty of Pharmacy, Universitas Airlangga, Indonesia. Prof. Dr. Bambang does not only deliver his lecture in Indonesia, but also abroad, where he became the visiting professor in several universities in Japan, Switzerland, and Germany. He is actively conducting his research in the Revenue Generating Unit Department of Natural Product Science, Faculty of Pharmacy, Airlangga University. He is also an avid reviewer in Majalah Pharmacy Airlangga and in Journal Obat Bahan Alam. He had been awarded with the Y Prize Travel Award to join in the American Society of Andrology Conference, promoting the scientificity of medicinal plant for male contraceptive. Now, given the development of synthetic chemicals for therapeutic use is by and large a random process that might result in serendipitous discovery, many scientists are now focused on the development of plant-derived drugs. As mentioned by one of our plenary speakers, Dr. Maya Gustina this morning, the Indonesian Food and Drug Authority has divided the preparation of medicinal plants into 3 categories: jamu, standardized herbal medicine, and phytopharmaca. So let us hear out from Prof. Dr. Bambang on the Indonesian medicinal plants for clinical studies, the needs, expectations, constraints, and risk. Please welcome him. Prof. Bambang?
Still mute, I think.
Prof. Bambang, are you still muted?
Saya unmute. Aku kan co-host kita. Sudah, Mbak.
Prof. Bambang, do you need any help? We can let the technicality to help.
Okay. Thank you very much, Ms. Moderator.
Thank you so much.
You hear me?
Yes, we can hear
Hello.
you clearly.
Okay. I want to share Yeah, thank you very much for giving me the opportunity to present my research work entitled The Journey of Gandarusa as Male Anti-Fertility. I've been conducted this research since 1987 and now enter the confirmatory clinical trial, and I already finished with the phase 1, phase 2, and phase 3. I would like to show the audience the result, including a preclinical evaluation and model of phase 1, phase 2, and phase 3 evaluation. This is a roadmap of the study, starting with the target selection and then lead discovery. We studied of medicinal chemistry, in vitro study, and in vivo study before clinical study. Kendarusa leaf have been used 4 years male contraceptive in Papua. This is medical. This is in Papua. One of the this plant grow well there and all part Indonesian from the 0 until 500 meter above sea level. Such practice benefit for Indonesian country since up the present time, my country still facing with a big problem in our demography. It is estimated that the population of Indonesia now reach number 260 million people with national population growth of the 1.3%. For example, each Java province and the population growth in Java is more than 1%, where the 47% of them are few family and have more than 2 children. This situation caused amongst other by the unawareness of the importance in improving the quality of life of children in the future. You see, and then here that this we have a national family program of keluarga berencana in Indonesia. Is a slow maintenance population in Indonesia. And then in ethnopharmacology, so is male infertility in Papua. And Justitia Kendarusa is Acanthaceae family. Grow well in Southeast Asia region and South of Asia, and growth in 1 until 500 above sea level is multiplied by steam occlusion. Traditionally, it's used as topically as a poultice for fever and rheumatism. Ethnomedicine study reveal that people of Papua have been using the plant as male contraceptive. Such a practice is widely conducted, especially among men who were not able to pay dower before marriage. They are allowed to live together as husband and wife, but are not allowed to have children. This research was conducted through study pharmacognosy, phytochemistry, toxicology, cultivation, preclinical and clinical study. Isolated compound was patented and now is the process of drug production, standardization of Simplicia or good drug and extract were conducted for quality control which is necessary for clinical study. In the regulation of the reproduction, anti-reproductive effect can be conducted with male and female volunteer with certain manipulated conditions, such as inhibition of ovum growth and ovulation, delay of fertile ovum migration, stop initial process of pregnancy and mid-semester abortion. Some conditions in male include spermatogenesis inhibition, inhibition of sperm maturation in the epididymis, spermatocyte, or disturbing the enzyme system of spermatozoa. This is a male contraception activity. This is a model manipulation how to inhibit spermatozoa function, include capacity, migration, binding, and penetration. The mechanism of sperm penetration is facilitated by three enzymes: hyaluronidase, ecrosin, and corona penetrating enzyme located in the acrosome of the spermatozoa. Its enzyme is connected to substrates located in the female ovum layers, the cumulus ovorus, corona radiata, and zona pellucida. In this region, the activity hyaluronidase shut down so that enable to digest cumulus ovorus substrate, resulted in the inhibition penetration. The natural compound with hyaluronidase inhibition activity, this is flavonoid, catechin, keratine, or flavanone, or flavonoid glycoside, flavonol, quercetin, and rhamnetine, or isoflavone, tectoridine, or tectorigenin. Preclinical evaluation. This is standardization of cultivation. Standardization is of medicinal plant standardized. This is a parameter of authentic of the plant, radioactive impurities, microbes, as contain volatile compound, macroscopic and microscopic, pesticides residue, marker compound, chromatogram profile, organoleptic evaluation, foreign material, and chromatogram profile. This is a plant of Justisia candarusa with the Acanthaceae family, the specific with the stem color with violet. This is a specific flower with this lancet type of leaf. The standardization of a crude drug, water soluble content 18.3, ethanol soluble content 6.9, water content is 11%, its content is 12.65, acid insoluble is content 1.93, volatile content is 0.065, total tannin is 1.3%, total flavonoid is 0.15, total calorie is 0.129%. This is a microscopic. This is a leaf of a candarusa with transversal section and cross section the leaf. After this is the standardization of extract. Water soluble content is 73%, drying loss is 61%, density of 1.2%, water content is 38%, its content is 10.3, and heavy metal impurities saying acubum and not contain acubum and cadmium. Gendarusin A is 0.84%. The general standard parameter of user Gendarusa leaf were equal with the general standard parameter of Materia Medica Indonesia. This is marker of Escherichia gendarusa Burman filius. This is epigenin derivative. We found in 2002. This is a major compound. It's a specific 12 of the flavonoid compound within the same molecule weight. This is flavonoid of gendarusa, gendarusin A, gendarusin B. This is we have 5 gendarusin A from the 12 gendarusin with the same molecule weight. This is a structure of alkali Escherichia gendarusa. It's also found with another register. It's also the alkaloid. And for the standardization of the raw material, we try with 7 location: Cibodas, Gempol, the Surabaya. Surabaya is a plant from the Papua, Makassar, Pacet, and Purwodadi. This is a scoreboard and a pin plot of the PCA. This is of in Gempol, Surabaya, and Papua is no different, but different from Purwodadi and Pacet. This is soil nutrition content is phosphorus, calcium, and cuprum. This separation between cluster is caused by different soil nutrition content phosphorus, calcium, and cuprum. The altitude, average temperature, average rainfall, climate type, and soil type had no effect. This is a random amplified polymorphism of DNA. This is from the 6 plant from the 3 location. This is of when DNA type and the profile like this. Antifertile mechanism. We research on with the infus, decreased meal rate serum testosterone, ethanol 60% in water, inhibit up to co-allanidase activity, ethyl acetate and butanol inhibit the mortality and viability of human spermatozoa by in vitro tests. And butanol fraction is thus 15 milligram per 20 gram body weight and inhibit the allanidase activity of mice. Water fraction, at least a single dose, this is inhibit the allanidase activity on Cumulus porphyris. Antiretroviral mechanism. Let's start the filtering after the continued treatment during one-time cycle of spermatozoa. Ethanol 60% at dose 15 milligram milliliter can be inhibited 50% human allanidase activity with competitive reversible. Water fraction at 15 milligram can inhibit 50% human allanidase activity with competitive reversible. Toxicity. We also have LD50, teratogenic report, no carcinogenics, the effect of electrolyte and biochemistry or toxicity data. As you know, in the Ibu Maya presentation from the National Food and Drug Control, Indonesian traditional medicine was divided in 3 categories: jamu, standardized, traditional medicine, and phytopharmaca. In our research, we focus in phytopharmaca. This is based on safety, proven benefit with a clinical test, already done standardized basic material, and fulfill the quality requirement. Pharmacology study, antibiotic study, is hyaluronic enzyme activity from the Gundarusa friction activity. This inhibit hyaluronic enzyme activity in bovine testis in vitro. This also activity is inhibit hyaluronic enzyme in human testis in vitro. Infus is a decreased male mice testosterone liver, where the friction is increased metacariocyte through hyaluronidase inhibition. This is a result of the how to decrease of the hyaluronidase. This is a negative control group with treated and positive control group with hesperidine. It is ideal. The product is reversible, so it's like the curve in this showing inhibition type of some with competitive reversible characterized with ethanol 60% fraction and water fraction of Gundarusa leaves. This is affect ethanol 60% fraction and water infection with hyaluronidase is a 3 to 1 cc in vitro. Effect of aspirin on epinephrine on hyaluronidase, this is like inhibitor competitive type. Osmotic swelling test is this is a method for how to know the integrity of membrane of spermatozoa. DNA profile, also we saw sort of mutilation compared with the negative of positive control, and reversibility test. Mice fertility could return to 100% normal after oral administration of Universitas Gundarusa. This is stop as per cycle. This research was done by oral administration. This is with scanning electron microscope. This is with water phase treatment. This is with ethanol fraction treatment. This is control mice. This is control mice after acrosome reaction. Effect mice spermatogenesis. Ethanol fractions of Gundarusa, dose 33 and 60 milligram, inhibit of phase 9 of mice spermatogenesis process. Water friction is did not effect of mice spermatogenesis process. This is a curve of the spermatogenesis phase in 9 stages. The increase of the cycles like this. The same, it's like the spermatogenesis between control and water phase. This is a histology of the testis, how to count spermatogenia, how to count spermatocyte and spermatid and spermatozoa. Effect spermatozoa motility, viability, concentration, and morphology. This is a water friction. This is did not affect motility, viability, and concentration of mice spermatozoa. Also the same with dichloromethane and methanol extract. In vitro test on mice. Here could inhibit mice fertilization in vitro. For example, This is of result of in vitro fertility stage 1 test on male mice. This is negative control group. This is treated group and positive control groups. All the treated group is unfertilization. How to collect the ovum? This is ovum bag. You see in the this is ovum in medium in 16. This is a spermatozoa, and in this picture, this is spermatozoa and ovum still intact. But in control is positive fertilization, and this is good normal mice ovum that has been fertilized into morula. In vitro threats and human effects spermatozoa motility and viability. This is we use ethyl acetate extract and butanol extract and dichloromethan extract and methanol extract and darussa. This could inhibit human spermatozoa motility and viability. How about effect in hyaluronidase enzyme activity in dispensing cumulus? Dichloromethan and methanol is dispensing human cumulus ovorus. This is a capsule process validation. Metabolic profiling during preparation of granule. This is a crude drug with sun drying, and then profile crude drug after free alkaloid. This extract and preparation of the granule. This is also the same. This is sun drying with different preparation, crude drug, free alkaloid, extract, and granule. This is metabolic profile comparison between laboratory and industrial scale. This is laboratory, industrial scale, and industrial scale. You can see the same profile both of the preparation. Clinical evaluation phase 1. Treatment is oral administration capsule containing Gendarusa dose 284 milligram and 213 milligram and placebo. Capsule were taken one daily for 1/2 period of human spermatocycle cycle for 108 days. This is a result of perimeter, hematology, urine complaint, liver function, lipid profile, kidney function, blood glucose, electrolyte. It is no significant change. Testosterone decrease. Electrocardiogram, this is a normal. Physical examination, blood pressure, normal, body weight increase, abdomen increase, other increasing appetite and libido. Semen analysis, no significant change. This is a semen analysis treatment group dose 264. This is concentration, morphology, and motility. Semen analysis result, the treatment groups dose 260 milligram. This is concentration, morphology, and motility. It's like this. Semen analysis result of placebo group. This is concentration, morphology, and also motility. Hyaluronate binding assay. This is also dosage 2, dosage 1, and placebo, like this. Bioavailability of Gendarusin A in human blood plasma. This is a blank plasma, chromatograph plasma with the Gendarusin A and chromatoplasma sample after treatment of Gendarusin A extract orally. It's like this. Bioavailability of Gendarusin A in human blood plasma. It's initial of the subject. and it's like this, content of gendarusin A and plasm. Okay. This availability of gendarusin A in human ejaculate plasm, chromatogram standard ejaculate that with standard. This is chromatogram gendarusin standard and chromatogram ejaculate that with gendarusin A standard. Where gendarusin in human ejaculate plasm, this is chromatogram ejaculate blank and chromatogram ejaculate sample. It's like this. We can found the gendarusin A. In human plasma, it's like this of the sample with initial ERM, day 100 each. Chromatogram is 100 each day, it's like this. This is recapitulation of elimination equation and half-life, like this, with the subject initial. This is average 0.19 and half-life 3.91. This is HPLC chromatogram of sample subject with initial DB. It's like this. This is conclusion. It's like this. Tusil Gendarusa was potential as male contraceptive material through enzymatic process which guarantee safety with a perimeter of non-teratogenic, non-carcinogenic, and non-mutagenic quality and efficacy based on WHO requirement on a contraception drug. This research was done in collaboration between National Family Planning Coordination Board, Laboratory in the Pharmacology and Phytochemistry École Pharmacie Genève, Lausanne, University de Geneva, Switzerland, and Hoshi University, Japan, Faculty of Medicine, Universitas Airlangga, Tropical Disease Center, Universitas Airlangga, this public hospital, Dr. Stomo, Materi Medika Batu, Faculty of Agriculture, Jember University, and Faculty of Science, Malaysia University, and pharmaceutical industry. The thing is, as multi-discipline members like this. Thank you. I beg.
Thank you so much. Thank you so much, Prof. Dr. Bambang Prajogo, for sharing with us on the availability and future direction of clinical studies on medicinal plant, especially in Justisia gendarusa. I hope I pronounced it correctly, in Indonesia. And all of us know that Indonesia has tremendous potential to be a herbal medicine producer due to its rich diversity in plants with medicinal properties. So it is such a pity, or actually, unfortunate for us to shift our conference to a webinar, because I think if we have the conference there, the committee in Indonesia would eventually allow us to see the beauty of what are the herbal medicine that is there in Indonesia. So now, without further ado, I would like to read on the question posted by our participant for both of our panelists. So the first question was posed by Dr. Rosa from USM to Prof. Bambang. Sorry. Given the work that you have done with Justisia gendarusa, what is your opinion if it is a standardized extract of a mixture of 100 to 1000 of compounds, how to determine the correct ADME profile? Prof. Bambang?
Okay, thank you very much. It's, this is for traditional medicine is consists of multi-compound. This is, we have one marker is gendarusin A. We can trace from the raw material and crude drug extract until preparation of product. So after use in oral in human, we can find the metabolite on urine, on blood of ejaculate. It's like this. So we think this can calculate half-life of the metabolites. This is like this. Okay.
So actually, this is a bit of like a clinical side of questions. Okay, did you have, out of curiosity, by Dr. Sarah from USM as well, do you have any idea on the acceptance level on the usage of contraceptives from men point of view? Because we normally heard about oral contraceptive for female. So have you ever did any study on to see the level of acceptance for male on this kind of intervention?
Okay. As you know, there is many contraceptive for women. 90% this is pill from hormonal or mechanical assessment for human, but for men just 3%. This is condom or vasectomy. So this is time for me to research and develop another contraception with non-hormonal, not hormonal contraceptive. So I think this is still safe with non-hormonal contraception. It's like this.
Oh, interesting. It's quite interesting. Okay, so now there's, can I just highlight the question to Dr. Fatima? So Dr. Fatima Tuzara, are you around? Can you hear me?
Yes, I can hear you.
Okay. So there's a question asked from Mrs. Retnosari. If we choose TNCM, is the cost covered by health insurance? I do hope that actually they were asking for Malaysia setting, because this is particularly for your question.
Okay, for Malaysia setting, basically for health insurance, there is none today, to my knowledge. Okay, but because all the services current that I shared with you, most of the services we have it in our public hospital, right? In our public hospital, the services are provided without charges. Okay, so every time this patient come, they just need to register for RM5, if I'm not mistaken now, and then they can get the medication and services in our public hospital. I hope that answered the question.
Okay, for the next question from Mrs. Hani. Okay, she's asking, there's actually two questions. So the first one is, if any, what are the strategies or policies in Malaysia that encourage doctors who usually prescribe chemical medicines to prescribe traditional medicines?
I answer this first. Okay, basically for the doctors in Malaysia, our policy, the doctors will prescribe all the modern medicine to the patient, and for traditional medicine, it will put under the traditional and complementary practitioner. This is what we're going to practice in the future. Meaning, if let's say currently we already have herbal therapy for chemotherapy as a adjuvant treatment for chemotherapy cancer patient. Okay, in this type of in this patient, whenever they want to start with the treatment, the medical doctors need to refer the patient to this clinic, and the what we call the medication or the herbal treatment will be prescribed by the TNCM practitioner, not the doctors. So we try to specialize the area in Malaysia.
Okay, and the next question from Hani. Who provide counseling services for safe and effective use of herbal medicines? Are there any pharmacists with specified requirements?
Okay, mostly in hospital, pharmacists will provide with the counseling. Okay, and as far for specific requirement, if let's say the pharmacist is under the TNCM units, they will send this pharmacist to maybe what we call it a short course that related to TNCM services.
Thank you.
Not really specific requirements.
Oh, okay. So from Bambang, there's one question actually. With regards to anti-malarial drugs, I'm sorry, it's like evening already. So developed from Artemisia annua and a therapeutic agent for the treatment of hepatitis, developed from the fruit of Fructus schisandrea, are the typical examples of successful drug discovery from herbal medicines, such as what you have done with Justicia gendarusa. From your point of view, how could this affect the principle of ecological ethics in preserving the biodiversity while trying to exploit the natural resources for drug discovery? Since I know that we should not wipe out any species arbitrarily and mess with the genes to create any transgenic crops.
Yeah, this is, I'm not focused in anti-malaria activity. I know this artemisinin from Artemisia annua, it's like Eusisia condarusa. This is a white plant. So for the industry, I think it's good for cultivation for the Condarusia condarusa, close with the industry. As you know, Condarusa is report with indomedicine in Papua is very far. This east of Indonesia, so it's a good idea after we to study about what is metabolic profile and characteristic of the plant. I think the same plant growth in Papua or in Java. I think it's good to cultivate in Java and to make supply to industry. It's like this, not for yes, like to distribute of Universitas Condarusa in Java. It's like this.
Okay. Well, thank you so much for Bambang, and thank you again also to Dr. Fatima to Zahra. So, well, I think that is all for the Q&A session. Thank you so much for our dear panelists for the fruitful and educational sharing. So, what I can say is that TNCM is an important and often underestimated part of healthcare, despite it is found in almost every country in the world, and the demand for its services is increasing. Traditional medicine of proven quality, safety, and efficacy contributes to the goal of ensuring that all people have access to care, and any countries now recognize the need to develop a cohesive and integrative approach to healthcare that allows the government, healthcare practitioners, and most importantly, the scientists for those who would use also the healthcare services, thus to assess TNCM in a safe, respectful, cost-efficient, and effective manner. Thus, we do hope there will be a global strategy to foster its appropriate integration, regulation, and supervision. So, ladies and gentlemen, we have come to an end our plenary session 2. We hope that it has been educational to everyone. However, this is not the end of the conference, as we still have the best poster and best presentation announcement and the closing ceremony of the ICPHS 2020. So, ladies and gentlemen, on behalf of the ICPHS 2020 committee, I would like to once again thank you all for the attention and participation. I would like to pass the next session to our Master of Ceremony. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Cik Nur Hafsan, for the fruitful discussion, and of the speakers as well. Thank you for the insightful material. And then, ladies and gentlemen, now we would like to give a token of appreciation as a certificate to all of the speakers for the second plenary session. And the certificate will be delivered by the Dean of School of Pharmaceutical Sciences, University, Sains, Malaysia. The first one coming to Dr. Fatima Zahra binti Abdul Aziz from School of Pharmaceutical Sciences, University, Sains, Malaysia. For the next one, to Professor Apoteker Bambang Prayogo Eko Wardoyo, Magister Science from Faculty of Pharmacy, Universitas Airlangga. Thank you so much, Prof. Matur nuwun.
Thank you.
And last but not least, we would like to give a token of appreciation to our beloved moderator, Cik Nur Hafsan Muhammad Hanafiah from University Sains Malaysia.
Thank you.
Thank you.
Excellencies, ladies and gentlemen, now we have reached the most anticipating agenda, the announcement of the best poster and the best presentation for the International Conference of Pharmacy and Health Sciences 2020. To all of the presenters, we believe each and every one of you have successfully delivered the idea about your research experiment, but still there is a winner. Now I would like to invite the representative from Scientific Committee of International Conference of Pharmacy and Health Sciences 2020 to announce the best poster and the best presentation. To Dr. Apoteker Suciati, the time is yours.
Thank you, MC, and thank you everyone for waiting. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to thank all participants of ICPHS 2020. Thank you for your contribution as poster presenters. And on behalf of the committee, I would also like to thank the judges for their hard work. We have received 266 abstracts, and today the judges have evaluated the poster and poster presentations. Each poster was evaluated by 2 judges from both Universitas Airlangga and University Sains Malaysia. There are 2 winners each for best poster and best presenters. It was a hard task for the judges to decide the winners since everyone have done a great job. There are 6 criteria for poster evaluation. First, background and objectives, methodology, results and discussion, language, conciseness, clear ideas and informative, creativity in poster design, and last but not least, overall poster presentations, which include balance of text, diagram, and arrangement. And the criteria for poster presentation are concise and clear delivery, engaging and interesting delivery, ability to answer questions, and time management. So, we have gathered the scores and the judges finally come up with the decision. So let me announce first for best poster. The second place with total score 42 is, I hope I pronounce it correctly, Ye Chuk Yap, poster code CP89, the title Therapeutic Drug Monitoring in Predicting Methotrexate-Induced Adverse Reaction in Patients with Rheumatoid Arthritis, Indicated or Not, from University Sains Malaysia. Congratulations. And for the first place is poster code number number CP12 with total score of 44 is Ayu Wulan Dwi Putri with the title The Effect of Intermediate Medication Review on Lifestyle Changes and Clinical Outcome in Patients with Diabetes. It's from Universitas Airlangga. Congratulations. And now, so we have 2 more winners for best presenter. The second place with total score 30, poster code CP61 is Ngah Kuan Cho. The title Health-Related Quality of Life and its association with socio-demographic, economic, and health status among HIV-positive patients on evaporations in northern Malaysia from University Sains Malaysia. And finally, the first place for best poster with total score 31 is almost excellent because the highest score is 32, is Kartika Diah Palupi, poster code NP31, the title Endophytic Fungi Inhabiting Physalis Angulata Plants, diversity, antioxidant, and antimicrobial activity of their ethyl acetate extract. From Indonesian Institute of Sciences or LIPI. So congratulations to all winners. We will send you the certificate by email and we need some details from you before we can send the award. So congratulations. Thank you. Assalamualaikum warahmatullahi wabarakatuh. Back to MC.
Thank you so much, Dr. Suciati. Once again, big round of applause to all of the winner for best poster and best presentation. So, ladies and gentlemen, we would like to remind all of presenter and also participants who do not fill out yet the questionnaire form, please do click on the link as follows. The committee will be sent to chat box. Please kindly filled out no later than 7 p.m. Western Indonesian time. So once again, we would like to encourage all of participants, all of the presenters, please kindly fill out the questionnaire form. Do click on the links as follows, as we mentioned in the chat box. And also, for your kind information, the full paper submission deadline is extended until 4th of November 2020. So once again, please kindly, may we have your attention please. For full paper submission, the deadline is extended until 4th November 2020. Now we almost reach the end of our conference. We will hear a closing remarks will be delivered by our co-chairman of International Conference of Pharmacy and Health Sciences 2020. Ladies and gentlemen, please welcome, coming from University Sains Malaysia, Dr. Siti Maisarah Sheikh Ghazi, PhD.
Thank you, Madam Master of Ceremony, for the invite. Professor Muhammad Nasir, Rector, Universitas Airlangga, Professor Datuk Dr. Ahmad Farhan Muhammad Sadullah, Deputy Vice Chancellor, University Sains Malaysia, Professor Junaidi Khatib, Dean, Faculty of Pharmacy, Universitas Airlangga, Prof. Dr. Habibah A. Wahab, Dean, School of Pharmaceutical Sciences, University Sains Malaysia, distinguished speakers and guests, ladies and gentlemen. Assalamualaikum warahmatullahi ta'ala wabarakatuh and good evening. Alhamdulillah, we are now at the end of our conference session. For all presenters and participants, thank you for your participation throughout the event. I really hope that you have enjoyed this event, had engaged with the works from the speakers and presenters, got to know each other, and have future plans to foster existing and future collaborations. And for the winners of Best Poster and Presenters, congratulations. I hope this event is one of the memorable one for you. With this, I would also like to thank all the committee members, both from UNAR and USM, for the blood, sweat, and tears in making this event possible, despite all challenges. My greatest appreciation goes to Faculty of Pharmacy, Universitas Airlangga, for being an excellent host for this virtual International Conference of Pharmacy and Health Sciences 2020 and for the opportunity to co-organize this event. I really hope that we will continue and enhance our relationship in the future through many collaborations to come. And finally, I would like to end my closing remark with a famous poem or pantun in Malay language. I believe Malaysians and Indonesians have inherited this poem from our ancestors. Here goes the poem, which simply means, till we meet again. Kalau ada sumur di ladang, boleh kita menumpang mandi. Kalau ada umur yang panjang, boleh kita berjumpa lagi. With that, I thank you. Assalamualaikum warahmatullahi ta'ala wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Siti Maisarah, for the wonderful closing remarks. Now, last but not least, we will hear a statement from our chairman of International Conference of Pharmacy and Health Sciences 2020. Ladies and gentlemen, Dr. Apoteker Ati Widyawaruyanti, Magister Sains.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Thanks to God Almighty, Finally, we have finished well all activities of the International Conference of Pharmacy and Health Sciences, ICPHS 2020, the third joint conference UNER USM, which carries the main theme, research acceleration in pharmacy and health sciences through the international collaboration. We would like to inform you that ICPHS 2020 was attended by 419 participants, including 249 poster presenters, 55 PADD workshop participants, and 216 HPTS workshop participants from Indonesia and Malaysia, as well as from several other countries such as Philippines, Nigeria, Canada, Australia, India, and Thailand. Ladies and gentlemen, although it is not easy for us, we were forced to hold this conference virtually due to the global pandemic COVID-19. However, I hope that during these 2 days, all of you enjoy this conference. In this joint conference, I believe all of us have obtained a fruitful discussion. We have learned a lot and gained valuable knowledge from the amazing speakers who share their expertise at the speaker station today, as well as from the experience and interactive speakers and tutors at the pre-conference workshop yesterday. On behalf of the committee, thank you to the speakers for delivering such important material so that all of us can gain our knowledge, especially in pharmacy and health sciences research field. I would like to appreciate to all of the presenters and participants who have participated actively to this ICPHS 2020 virtual conference. I'm certain that every presenter has presented the application of their research work. I sincerely hope that through your presentation and discussion afterwards, all of us can contribute to solving the problem in our own countries, even though the contribution may be small, but it's very valuable. We also like to congratulate to the winner, the participant who winner the best presenter and the best poster award. To all participants, I would like to remind you, even though sorry, even though this is the last day of the conference, there is some work that we should do regarding your full text paper submission. For the further information, our scientific program committee will inform you later. Ladies and gentlemen, my deepest gratitude to Dean of Faculty of Pharmacy, Universitas Airlangga, and Dean of School of Pharmaceutical Sciences, University Sains, Malaysia, for supporting the facilities that make the committee accessible to organize this conference. Finally, I will also want to express my deepest gratitude and appreciation to all ICPHS 2020 committees, both from UNER and from USM, who have worked hard for organizing this conference and ensuring that it proceeded smoothly. Thank you for the great cooperation between us. And I apologize to all speakers, participants, and presenters for any inconvenience that you might experience during this conference. And I do hope that we can meet again in the next fourth international joint conference, UNER USA. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Once again, big round of applause to all of the organizing committee of this international conference. Thank you so much. Ladies and gentlemen, finally we've come to the end of our agenda, International Conference of Pharmacy and Health Sciences 2020. On behalf of the organizing committee, we would like to give thank you for making time to join us here today. We would like to extend our gratitude to all the participants and presenters in making this conference a success. It has been our pleasure to host this event, and we look forward to seeing you at the next conference. Stay healthy, everyone, and we wish you have a pleasant day. Wassalamualaikum warahmatullahi wabarakatuh. Big round of applause to all of you. Thank you so much. We would like to invite all of the organizing committee, the people behind the scene. Please come forward to all of the hard-working committee of the International Conference, Pharmacy and Health Sciences. And we have a special guest, our beloved Dean, Faculty of Pharmacy, Universitas Airlangga. Also with us here this very afternoon. Everyone, please come join also. Okay. Okay, thank you so much, everyone. See you again in the next conference. Stay healthy. Bye-bye.